So, good morning, everyone. Welcome back to our next presentation. This is the fifth in the series, if you've been keeping track. And in this presentation, obviously, we're going to be covering Chapter 5, which is all about the really large molecules that we find in cells. In fact, these are the molecules that make up that missing 30% we haven't talked about yet. Remember, 70% of our bodies are made up of water molecules. And then the remaining 30% are those that are built around carbon skeletons. And in this chapter, we're going to focus very specifically on those molecules that meet that criteria. At the heart of all of these molecules is going to be some form of carbon skeleton. And to that is going to be attached the functional groups that we were talking about in the last presentation. But what we will also discover is that of all of these large macromolecules, when we look at the huge diversity of macromolecules that are present in organisms, we can divide them up into just 4 different classes of molecules that we would describe as being macromolecules. Of those 4 different classes, 3 belong to a group of molecules that we would define as polymers, or we would describe as polymers. Polymers are really large molecules that are assembled or created by assembling together lots of little molecules known as monomers. And so 3 of the very large or classes of large molecules that we find in cells are described as polymers. The 4th class, which is not strictly a polymer, but is still a very large molecule nonetheless, is assembled from molecules that are themselves pretty large to begin with. And so really don't qualify as polymers, nor do they consist of lots of little molecules strung together in long chains, which is strictly what a polymer looks like. But we consider them along with the polymer classes for 2 reasons. Number one, they're all large molecules, so they meet the criteria of macromolecules. And number 2, it turns out that the chemical reactions that assemble and disassemble all 4 classes of macromolecules are exactly the same, whether they are one of the 3 polymer classes or whether they belong to the 4th outlier class that is not actually polymers, but is nonetheless a large molecule in its own right. And so the 3 classes of, or the 4 classes of molecules that we are actually referring to in this group are, and you probably recognize some of these, perhaps all of them, are carbohydrates, which many of you know as sugars, lipids, which most of you would refer to as fats, proteins, and the nucleic acids. The 3 polymer classes are carbohydrates, proteins, and nucleic acids, and the lipids are the 4th class that are not strictly polymers, but are nonetheless big molecules. And as I said, chemical reactions that assemble and disassemble them are exactly the same as those that assemble and disassemble the polymer classes. So it is kind of appropriate that we consider all of these together. But before we look at each class independently, let's talk a little bit about what is meant by polymer and monomer, for that matter, why this matters as we consider these macromolecules, and then take a look at the 2 chemical reactions that are involved in assembly and disassembly. So, because remember again, these chemical reactions are uniform across all 4 classes of macromolecules, and we will see specifically how those chemical reactions are used in the assembly and disassembly of each of the 4 classes of macromolecules as we begin our discussion of each of them. So let's move on and take a look at what is meant by a monomer and a polymer. So strictly speaking, a polymer is a very long molecule or a large molecule that's created by linking together lots of small molecules, which we refer to as monomers, in a daisy chain. So it's a long chain-like molecule built out of the monomers. Each of the 3 classes of polymers that we recognize as being important in life, the carbohydrates, the proteins, and the nucleic acids, have as a consequence their own unique group of monomers that are assembled together to create them. So if we fire up the pen here and let's start making a few notes about the carbohydrates, the proteins, and the nucleic acids. So carbohydrates, let's put AKA sugars, because that may be what you know them as. And the monomers are called monosaccharides. So you recognize the prefix mono. You may not recognize the word saccharide, however, but that's the Greek word for sugar. So the monomers are single sugars, and we'll look at a sample of monosaccharides that are assembled into the large polymer carbohydrates that are of interest to us. We should not lose sight of the fact that some of the monosaccharides are also important, as you'll see as we move forwards with the semester. But we'll also look at the long chain or polymer carbohydrates as well, because some of those turn out to be structurally important. In terms of proteins, well, sometimes we use the word polypeptide. When we talk about the proteins a little bit later this morning, or in this presentation, it is morning as I'm recording this, hence why I keep saying this morning. We'll look at the distinction between polypeptide and protein, because while a lot of people mistakenly use the words interchangeably, they really don't mean the same thing. There is an important difference between the term polypeptide and the term protein, and you really do need to know the difference. So when we talk about proteins in more detail later in the presentation, I'll explain what the distinction is. The monomer here, and we've heard about these before, is the amino acids. And I actually drew a picture for you, or showed you an illustration in the last presentation of what an amino acid looks like, the structural features of amino acid. We'll revisit that in this presentation, but we'll go into more depth about what an amino acid is and what the significance of understanding something about the amino acids is in terms of understanding proteins and polypeptides. Let me just move the text over here so I can use the full depth of the slide. So nucleic acids, oops. I'm going to fire up the pen every time I do something like that. So nucleic acids, the monomer here. Is the nucleotides. Let's erase that and do it a little neater. So the nucleotides and the polymers are the DNA and RNA. And most of you have probably at least heard of these. If you don't know very much about them, that's okay at this point. We're going to talk a lot about them this morning in terms of basic structure. And then when we get to chapter 16 and 17, we're going to talk even more about structure and function. But there's some important information that I want to put in front of you now, because it helps when we get to chapter 16 and 17 if we build a solid foundation in terms of your understanding of what the polymer nucleic acids are, plus the monomers, in this chapter, because once we get to chapter 16 and 17, we can build on that solid foundation to improve your knowledge of DNA and RNA and how they function in cells. So we will spend a little bit of time in this presentation talking about the structure of the DNA molecule and the RNA molecule, and we'll talk a little bit about their function as a consequence of that as well. So these are our three polymer classes, and then of course we could add in here, if we want to put another one, you could put in here, and we'll just put it in here, lipids. aka fats, and we'll just go large, but not polymers. So that we've got the four classes of macromolecules listed here together. The three polymer classes, carbohydrates, proteins, nucleic acids. The fourth class of macromolecules, the lipids, not polymers, but they are very large nonetheless. So when we start thinking in terms of polymers, we have to think in terms of how do we build them from the small molecules, and also how do we disassemble them again from the polymer form back to the small molecules, the monomer form. And it turns out that there are two chemical reactions involved. One is referred to these days as a dehydration synthesis. Sometimes it has been referred to as a condensation reaction. So I'm just going to put in here preferred name. It's now a dehydration. I probably need to edit this slide to change that out, because there is a change in terms of what is the preferred name. It used to be condensation reaction because of the nature of the reaction and how it takes place. But there seems to be a move now to prefer the name dehydration reaction. They're talking about the same thing, and you may, in some of your external reading, or if you look at some of the resources that I've posted in the resource page, some of the YouTube videos, may see the term condensation reaction used rather than dehydration reaction. They are talking about the same thing. And that is the type of reaction that is used to assemble the polymers from the monomers. In other words, it links the monomers together to create the polymers. The reversal of that is called hydrolysis. So it basically does the opposite to what the dehydration reaction does. So if a dehydration reaction, as the note says here, removes the functional equivalent of water in the process of linking the monomers together, then hydrolysis splits them by adding back the functional equivalent of the water. So you're going to see a hydrogen ion and a hydroxyl group, or a hydroxide ion, removed as part of a dehydration reaction. There's your functional equivalent of a water molecule. And when you do the hydrolysis reaction, you're going to add back a hydrogen ion and a hydroxide ion as the functional equivalent of water being added back to split the polymer into its monomer components again. So as always, a diagram is worth more than just a verbal description or a written description. So let's take a look at the diagram that illustrates how this takes place. So here is an existing polymer. Notice that this then we have a hydrogen. That it depends on the type of monomers and the polymer that you're creating as to which of the two groups is on the polymer, which of the two groups is on the monomer. In this illustration, we've got the hydrogen on the polymer, and we've got the hydroxide or hydroxyl group on the monomer that is going to be linked. So we take these two, I think I'm going to change this to red. So we're going to take these two out. That becomes our water molecule. And in the process, we create a new covalent bond that links the polymers together. Let's just put up here, breaks existing. So it breaks the existing covalent bonds that are holding the hydrogen and the hydroxyl group to their respective molecules. That gives you the opportunity to create new covalent bonds. One of the new covalent bonds creates the water molecule. The other new covalent bond attaches the monomer to the polymer, increasing the length of the polymer. So that's the condensation reaction or dehydration reaction. Either name works. The reversal of that is hydrolysis. And if we look at the two components, hydro refers to water, and lysis to split. So a hydrolysis reaction literally splits the covalent bond with water. So again, we're going to break existing covalent bonds. We're going to break this covalent bond, and we're going to break the covalent bond of the water molecule. And that's going to then form two new covalent bonds, the covalent bond that links the hydrogen to the now shortened polymer, and a new covalent bond that's going to link the hydroxyl group to the now separated monomer. But what we've done is split the covalent bond by adding these two functional groups to the relevant ends of the shortened polymer and the now liberated monomer. So we can consider these as the key reactions that we're going to see as we start talking about both the three polymer classes, but also as we start talking about the lipids, the fats as well. Because remember, these reactions are identical, irrespective of whether we're looking at the polymer classes or whether we're looking at the lipids. It's these reactions that are used to assemble and disassemble all of the macromolecules, irrespective of whether they are polymers or not. So that's the important thing. Where we start to encounter diversity and where we start to understand why life is diverse as it is because even though, for instance, if we're looking at proteins, all proteins are built out of a set of 20 common amino acids that are pretty much universal across all life forms. So if all the proteins in all of the organisms on the planet are built out of just the same set of 20 amino acids, how does that lead to diversity of life? Well, there are essentially an infinite number of ways of assembling those amino acids together to form proteins. And it turns out that each organism may share some proteins in common with other organisms, and this is not unusual, but every type of organism also has its own unique types of proteins. And that's what allows for the diversity of life. Likewise with DNA. If DNA is the molecule of inheritance and carries the genetic information, then we would expect that each species would have its own unique form of DNA molecule. Again, DNA is built out of just 4 monomers, the 4 nucleotides. But there is essentially an infinite number of ways of assembling those nucleotides together to create information or to represent a source of information. And not surprisingly, there is a lot of difference between DNA from unrelated species, indicating that it is possible to explain the diversity of life by looking at the differences in the DNA. Something that we've only really been able to do in the last 30 years or so in great detail. But nonetheless, it really does help us understand why life is so diverse. And just to reinforce that, when we look at species that are really closely related to each other, we find only a small number of differences. So if we compare humans with chimpanzees, our closest living relative, there are only 6 differences. But they're not really at the genetic level per se, because we share pretty much the same genes with chimpanzees. It's how those genes are regulated. So the 6 differences are all about controlling how those genes are utilized, not the differences in the genes themselves. And that's what really makes the difference between humans and chimpanzees as 2 very, very closely related species. And so we can Still, when we think in terms of the details, understand the diversity of life by simply understanding the diversity of polymers, even though the polymers seem to be restricted at first glance because there's only a limited number of monomers for each type of polymer, given that there are, at least for proteins and DNA and to some degree the carbohydrates as well, an unlimited number of ways of linking them together, then suddenly you can explain the diversity by those unlimited number of possibilities. So we can really look at the polymers and get a really good handle on where the diversity of life comes from. We don't have to think in terms of, well, it's a restricted number of monomers, that's going to restrict the number of polymers. That's really not a good correlation anymore. Having a limited number of monomers does not restrict the number of polymers you can create. So that's basically what this slide is talking about, just talking about the variety of polymers, which I've already talked about. So let's stop for a minute, take a very quick break to check in on a concept and see how you would go about answering this question. So it may be a little bit early for you to go and take a proper break, but let's pause for about 30 seconds while you consider this question. How many water molecules are required to hydrolyze a polymer chain 10 monomers long? So let's take a look at this question and see how you answered this question. So I'm going to go out on a limb here and say that a number of you probably jumped on the number 10 here and said, if there are 10 monomers, then I'm going to need 10 water molecules to completely hydrolyze this polymer. And at first glance, you would think that that would be logical. But you're wrong if that's the conclusion that you came to. And let me draw a little diagram to illustrate this, why 10 is the wrong number. So if we've got 10 monomers linked together, and let me turn the black back on. So I'm just going to use numbers to represent the monomers. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And then I'm going to represent the covalent bonds that are linking them together with black dashes. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9. If you've got 10 monomers in the chain, there's only 9 bonds that are holding those monomers together. So if you're going to hydrolyze all of the bonds that are linking the monomers together, you're not going to need 10 water molecules, you need 9 water molecules. And that's something that you can remember, because if you see any question on an exam or in a practice module or something like that that asks you about the number of water molecules to hydrolyze a monomer chain, it's always going to be 1 less than the number of monomers in the polymer. Okay, let's move on and let's start talking about the first group of polymers, the first group of macromolecules, the carbohydrates, which unfortunately have got a bad name amongst people who are looking for alternative diets, because as you might know, there are lots of low-carb diets out there, and you're probably aware of a number of them. The problem with going on a low-carb diet is that your body uses carbohydrates, and particularly one carbohydrate, glucose, as its primary cellular food, its primary cellular energy source. And very particularly, your brain is very dependent on glucose. And the only way that you can get glucose efficiently is in your diet. So cutting carbohydrates out of your diet is not really what you want to do. The reason why, however, carbohydrates have got a bad name is because they are a ready source of energy. And if you eat a lot of refined carbohydrates, so table sugar like sucrose, or some of the simple sweeteners that are used that are basically just solutions of lots of glucoses and maybe fructose or something like that, or some of the modified sugars that food manufacturers are now trying to sneak into food in a way that you might not recognize them, or using alternative names for table sugar. So if you see written on an ingredient list the term dried cane juice, don't fall for that one. Dried cane juice is just sucrose, table sugar, because that's where our table sugar comes from, sugar cane. The juice pressed out of sugar cane is allowed to dehydrate. We evaporate the water out of it and the sugar crystallizes out of it. It's usually in a raw, dark brown form because it's still got molasses in there. To get the white sugar, we simply spin the molasses out, redissolve it, and keep spinning the molasses out until we've got it all out. And what we're left with is the white sugar that we're all familiar with from the supermarket. Ironically, they're now selling a lot of brown sugar in supermarkets, and what they're doing is, instead of keeping the molasses in the sugar, they're actually taking white sugar and adding molasses back to it to create brown sugar. Yeah, it doesn't seem really logical, does it? But that's the way it's being done. That's the really big issue, is not so much eating carbohydrates, it's eating the wrong sort of carbohydrates. So if you're eating lots of simple carbohydrates, the only thing that they give you is a lot of energy, a lot of calories. And yes, we should restrict the amount of simple sugar we take in, but we shouldn't restrict the number or the amount of complex carbohydrates. And we'll talk about some complex carbohydrates a little bit later in this section as we talk about the different types of carbohydrates. But let me stop for just a minute and ask another question. And of course, I'm not going to hear your answer to this, but it's a question that's worth thinking about. How much sugar do you think on average you consume every year? So you've probably got a number in mind. Let me assure you that you probably, unless you're unusual and have a real handle on how much sugar you really do consume, have underestimated, because we know that the average American consumes around 140 pounds of sugar per year. Some people will say 100 pounds, but the consensus from all the literature that I've found is that the number is close to 140 pounds per person per year. And if you want to know what that constitutes, the old bags of sugar that they used to sell in the supermarket, they've actually reduced the size of the bags a little bit, probably so the price doesn't go up too much on a bag of sugar. The largest bag of sugar that you could buy in the supermarket previously was a 5-pound bag. And so if you wanted to stack a number of those bags to get close to the 140 pounds, you'd have to stack 28 bags to get the 140 pounds. That's a lot of bags of sugar. Doesn't matter how you look at it, that's a lot of bags of sugar. And no one in their right mind would consider consuming that amount of sugar in one hit, because you would get sick after a while. But why we consume that much sugar is because sugar is added into so many things where we don't expect to find it. And unless we're paying attention to the ingredients list on our foods, we're not even aware of how much sugar we're consuming. So if you look at the typical peanut butter, it's got sugar in it, and it's got added sugar in it. It's not just the carbohydrate in the peanuts, which is probably healthy carbohydrate. It's the added sugar that's put in there. in the form of dextrose, typically. In fact, if you want to look for sugar, you can identify sugar So if you see something on the nutrition label of your favorite food that ends in OSC, even if you don't recognize the name, that's a sugar. I mean, take a look at the names of the sugars we've got up here: ribose, deoxyribose, glucose, sucrose, lactose, maltose. They're all sugars. They all end in OSE. Dextrose, another name for glucose, ends in OSE. The other thing that you might see So the two that you're most likely to see here that end in OL, these sugar alcohols, are sorbitol and mannitol. Those are the two that typically are used in food as alternative sweeteners. And they're just as energy rich as sugars. And these are all simple sugars. These are all monosaccharides or derivatives of monosaccharides. And they're the ones that usually are the ones that are cheap, easy to use as sweeteners, and contain a lot of calories and nothing much else, because there's no other nutritional value in a sugar other than calories. They don't bring any other nutritional value with them. So these are the ones to look out for. So these are some typical monosaccharides that we'll encounter as we progress through the semester. So ribose and deoxyribose are the sugars that give RNA and DNA part of their name respectively. So RNA is ribose nucleic acid, DNA is deoxyribose nucleic acid. Glucose is blood sugar. I'll put a note here, this is the blood sugar. And this is what our cells prefer as an energy source because it's easily broken down to release energy, and it releases a lot of energy through cellular respiration. And this is the food that our brain preferentially wants to use because it's one of the few energy molecules that can easily cross what is called the blood-brain barrier. Those of you who are going into the health sciences in some form, whether you're going to medical school or nursing school, allied health sciences, if you do our AMP course or some equivalent, you will learn about the blood-brain barrier. It's quite an important part of your understanding of the physiology of the human body because it's what determines what can cross out of the bloodstream into the brain and what can't. And glucose is one of those molecules that can easily cross the blood-brain barrier because it's what our brain uses as its fuel. And even though our brain accounts for less than 10% of our body mass, it actually utilizes close to 40% of our energy. So providing it with the right source of energy is really, really important because it is an energy hog, but it only uses very specific types of energy. So this is another reason why we don't really want to cut carbohydrate out of our diet. Our body is flexible enough to be able to make glucose from other sources. So if you eat a lot of fat, if you eat a lot of protein and not a lot of carbohydrate, your body can convert some of that to glucose, and it will definitely do that because it really does need that sugar as an energy source. But it doesn't do it terribly efficiently, and it doesn't do it very effectively either. And so your body is constantly going to run in carbohydrate deficit. And don't be surprised if your body plays tricks on you and craves sugar so much that you actually give in and eat sugar. This is one of the things that we always notice with these very restrictive diets, like the low carb diets, like the high protein diets, like the high fat diets. Anything that messes with the ratio of carbohydrate, fat, and protein that our body prefers and emphasizes one to the exclusion of the others usually winds up failing, because your body knows what it needs, and sooner or later it's going to rebel against your diet. And so people who follow these low carb diets or the high fat diets, all of these so-called fad diets, almost invariably wind up falling off the wagon, so to speak, quitting the diet because they just can't keep going. And there's no value in a diet if you can't continue to follow it, because when you make changes in your eating patterns, that's got to be something that's permanent. It's got to be something that's doable. It's got to be something that you are happy to follow and your body is happy to follow for the rest of your life, particularly if you're trying to manage your weight and your body fat content. Because every time you change your eating habits, your body will adapt to those changes, and it will fuel itself up, and it will, and particularly if you go back to your old eating patterns, because you've just got tired of the diet that you're on, not only will it restore its former self, add back the fat and so forth, but it will become harder and tougher if you decide to try and lose it again. Every time you go up and down, what's called yo-yo dieting, every time you go up and down, it just becomes harder and harder and harder to make the changes again. Your body may be flexible, but it can also be quite inflexible at times when it comes to body composition. Just bear that in mind as you consider how your eating habits and how you might want to modify them over the course of your life. Focus on doing things that you can sustain, not something that you do for a couple of weeks and then return to your old habits again. Because every time you go back to your old habits, your body returns to its previous form, and it's more than willing to do so. And so focusing on one macronutrient like carbohydrates or protein or fats to the exclusion of the others, and adjusting your diet to emphasize or de-emphasize one to the exclusion of the others, is simply putting your body in a stress zone that it's not happy with, and sooner or later, you know what's going to happen. You're going to go back to your old patterns again, because none of those diets are sustainable. This is the history of those diets. They go through cycles. None of them are new. The Atkins diet, the South Beach diet, all the other types of diets, none of those are new. They've been known for about 150 years through various different names. If they had been successful 150 years ago, we would not be renaming them and quote unquote rediscovering them every 50 years or so, because people would say, this is a diet for life. These are not diets for life. If you can't sustain them for the whole of your life, if you can only sustain them for a few months and then you fall off it again, that's not a diet for life. And the history of all of these diets is they are unsustainable as a diet for the course of a person's life. Because sooner or later, your body says, I can't do this anymore. Just can't do it anymore. Anyway, I'll stop talking about diets for a little while. We'll talk a little bit more about a couple of things related to diet, particularly when we get to fats, which is the next group, and a little bit when we get to proteins, but mostly when we get to fats, because there are some issues there with fats that we need to pay attention to in terms of your intake of fats, what's healthy and what's not. So carbohydrates. So here are an example of monosaccharides that we will encounter over the course of the semester. So these 3 carbon sugars, glyceraldehyde and dihydroxyacetone, are the breakdown products of glucose and fructose in cellular respiration. So glucose is converted to fructose, which in turn breaks down to a molecule of glyceraldehyde and a molecule of dihydroxyacetone during the first part of cellular respiration, called glycolysis. And so you're going to encounter these 2 sugars. Now notice that the carboxyl group on these 2 sugars is highlighted. The reason why is because these 2 represent structural isomers. Same number of carbons, hydrogen and oxygens, just arranged into 2 different structures. So these are structural isomers, and they are interchangeable. There is an enzyme that converts glyceraldehyde into dihydroxyacetone and back again, called isomerase. And that is also a part of glycolysis, which we'll get to when we get into chapter 9. But this identifies now that there are 2 different classes of sugars that are important in life. So-called aldehyde sugars, because the carboxyl group is terminal, which you should remember from last week is called an aldehyde carboxyl group. So these are called aldehyde sugars or aldoses. And then you've got dihydroxyacetone, where the carboxyl group is in the middle of the chain, non-terminal. And so you should recall from last week that this is called a ketone. And so this is a member of the ketone sugar class, or ketoses. And you can see that the remaining sugars down here also are divided into those 2 groups. So ribose is an aldehyde sugar. Its structural isomer, ribulose, is a ketone sugar or a ketose. Ribose, well, we've talked about that. That's in RNA, and deoxyribose is in DNA. Ribulose we're going to encounter in photosynthesis. Glucose and fructose I've already mentioned in cell respiration. Glucose is a 6-carbon aldehyde sugar or aldose. Fructose is its structural isomer cousin, and it is a ketose. But then notice that we've also highlighted, oops, go back, this carbon in glucose and another hexose called galactose. Both of them are aldoses, so these can't be structural isomers. So why highlight this? Well, notice the orientation of the hydroxyl group and the hydrogen on this carbon. They're on opposite sides between the 2 molecules. And if we take a look at the carbons themselves, what we discover is that they are in fact chiral carbons or asymmetric, because there are 4 different things attached. So this one is obvious and this one is obvious. But then here is number 3. And by now you should be guessing this is number 4. And if we do the same thing for galactose, and I'll do it in red, is number 1, number 2, number 3, and number 4. So these are asymmetric carbons or chiral carbons, or as you'll see a little bit later, we sometimes refer to these as alpha carbons as well. The point here is there are 4 different things attached to this, and if you recall from last week, if you have 4 different things attached to a carbon, you have the potential for mirror image isomers or enantiomers. And so glucose and galactose are actually enantiomers of each other, left-handed and right-handed mirror image isomers of each other. And they are somewhat interchangeable via a chemical reaction. There is, in fact, a whole family of isomers that are associated of mirror image isomers that are associated with the basic hexose conformation, because if you go down through the carbons, what you'll come to realize is that this carbon is asymmetric, this carbon is asymmetric. We've already said that this carbon is asymmetric. This one is asymmetric. This one is not, because it's got 2 hydrogens attached to it. So it's only got 3 different things, not 4 different things. This one is not asymmetric because, again, it's only got 3 different things, not 4 different things. But of the 6 carbons in glucose and likewise with galactose, 4 of them are asymmetric and therefore can form mirror image isomers and enantiomers. So there's a whole family of enantiomers based on this basic aldose 6-carbon structure. And likewise with fructose, these 2 carbons are not asymmetric, and this carbon is not asymmetric, but this one, and this one are. And so there's the possibility of enantiomers around these 3 carbons, and we know of those isomers as well. Some of these isomers exist in nature, some of them are only known from the laboratory, but the point here is there is a wide variety of enantiomers possible because of these asymmetric carbons. And so immediately we start to see a greater diversity of molecules built around similar structures. And most of these are known from plants. Plants are the ultimate organic chemists. And they have the capacity to produce this wide range of diverse organic molecules. Animals can as well, but not quite to the same degree as plants. Bacteria are right up there with plants in terms of the variety of organic molecules that they can produce. But plants are the ultimate organic chemists because they build all of their organic molecules from carbon dioxide and water using energy from the sun via photosynthesis. That's their starting point. So they are really the ultimate organic chemists on the planet. So immediately we start to see where the diversity of life can come from by the diversity of molecules that can be produced. And we're starting to get a handle on that diversity. So one of the things that we have recognized is that not only do we have these linear forms of the carbohydrates, we also have a circular form. And specifically, the circular form is important because these are what are going to be linked together to form polymers. And while you don't need to know exactly how the linear form forms up, if you take organic chemistry, which some of you will if you're going for a biology degree, I mean, you will take organic chemistry if you're going after a biology degree, you'll learn how the circularization takes place because it's important for organic chemistry. Here, I'm not really interested in you understanding how it takes place, just that it does take place and that it has consequences for how the polymers form up. So this is the circularization of glucose. And you'll notice again that we are numbering the carbons in the carbon chain, and we're following the IUPAC rules, which says that you number from the carbon that contains the most interesting or unique feature. So the unique feature in glucose is the carbonyl group attached to carbon number 1. So we go 1, 2, 3, 4, 5, 6. And the circularization is between carbon number 1 and the oxygen on the hydroxyl group on carbon number 5. That puts carbon number 6 outside the ring. Then when we draw this structure, we typically leave out the carbons. And this is a very unique thing, or a very common thing, should I say, that we use in organic chemistry. Wherever we have an intersection of lines in a diagram of a molecule, that intersection represents a carbon unless we're told otherwise. So here, where we're looking at the ring form of the sugar, each of these points, and they've put a number against them, represents a carbon atom, even though we don't have it diagrammed in there. And so this is the circular form of glucose. And it's actually one of 2 circular forms of glucose, as you'll discover a little bit later. So how do we go ahead and link these together to form polymers? If it's this ring form that's the key, how do we go ahead and form the polymers? So the first type of polymer is something called an oligosaccharide. And this is usually, I've got it in red, so let me turn it back to black. So we usually use the term oligosaccharide for monosaccharide chains anywhere from 2 to 5 monosaccharides in length. But the most common oligosaccharides that you'll encounter in a biology forum are disaccharides. So table sugar, sucrose is a disaccharide, milk sugar, lactose is a disaccharide, and cereal sugar, the sugar that's found in cereal grains called maltose, is also a disaccharide. Glucose is a disaccharide formed from glucose and fructose. Lactose is glucose and galactose. Maltose is 2 glucoses. So these are the common disaccharides that we encounter in a biology forum. So how do these disaccharides get formed? So here's how we form maltose. Maltose is formed from 2 glucose molecules. So here is the circular form of glucose. You should recognize that. Remember, this is a dehydration reaction. So we're going to take a hydroxyl and a hydrogen. That gives us our functional equivalent of water. That then creates an oxygen oxygen bridge between the 2 glucose molecules. And this is referred to as a glycosidic linkage. So if you see this oxygen bridge between 2 monosaccharides, you've got a glycosidic linkage. The type of glycosidic linkage is determined by which of the carbons in each of the 2 sugars is being bridged. So in this case, it's the number 1 carbon of this glucose and the number 4 carbon of the second glucose. So this is referred to as a 1,4 glycosidic linkage. And the vast majority of organisms are able to break this linkage. In other words, they can digest maltose. And so maltose can function as a source of glucose and therefore as a source of energy. Sucrose, table sugar, which is formed by plants as a way of transporting sugars from where it's produced in leaves to various other parts of the plant where it's going to be used as an energy source. So all plants make sucrose. It's just that sugarcane and sugar beets make so much sugar that they store it. In the case of sugarcane, in the cane itself, in the case of sugar beets, in the root, and that's where we get our sugar from. So sugarcane is used pretty much all around the world as a source of sugar, originated in India, but has been transported all around the world and is grown in a lot of countries these days. And sugar beets, which are used in Europe as an alternative way of obtaining sugar, because sugarcane won't grow in cold climates, sugar beets will. But basically, you grind up the plant material, squeeze the juice out of it, allow the water to evaporate, and what you're left behind with is crystallized sugar. Then you can purify it. As I said, with our sugar, we spin the molasses out of it to get the nice white sugar crystals that we buy from the supermarket. So that's table sugar, otherwise known as sucrose. And it's a molecule of glucose and a molecule of fructose linked together. Now, both of these are hexoses, but remember, glucose is an aldose, an aldehyde sugar, whereas fructose is a ketose. And that's the reason why fructose has a slightly different form of ring structure than glucose. So if we jump back for a minute, back here, here's where the carbonyl group is in fructose. And so the link is going to be from here down to here. That's when it circularizes. That's where the link is. So this carbon and this carbon, oops, maybe I should put the numbers on those because they actually numbered. So this is actually given, this is carbon number 1 and this is carbon number 6. So both of those carbons are going to lie outside the 5-sided ring that is the circular form of fructose. So if you're wondering why it looks different, that's why. Is it important that you know that in detail? No, but I just want to point that out so that you understand. But that's going to change the way we look at this in terms of the type of glycosidic linkage that is formed as well. So again, it's a dehydration reaction. We take the hydroxyl and the hydrogen, there's our water. We form the oxygen bridge, but this time it's between carbon number 1 on the glucose and carbon number 2 on the fructose, 1, 2, 3, 4, 5, and 6. And so this is known as a 1,2 glycosidic linkage. And there is no organism on the planet that is incapable of breaking this glycosidic linkage, because this is the way plants transport excess sugar around inside the plant. It's important that organisms be able to break glucose down to get access to the energy in plants when they either eat the plants or eat the sugar in some form. So breaking down sucrose, pretty much every organism on the planet can do that. It can break the 1,2 glycosidic linkage. But that's the reason why we need to stay away from glucose. We can break sucrose. We can break this. We can get access to all the calories in here, but there's not much else present. And by the way, those of you saying, but I don't eat table sugar, I eat honey. Honey is just a very concentrated sugar syrup, and it doesn't have a whole lot of other nutritional benefit. It is basically a very concentrated solution of sucrose produced by bees. And if you really want to know what it is, it's bee spit. Sounds really attractive when you think about it that way, but that's really what it is. The nectar is processed by bees in the hive and allowed to then dehydrate, and what you get left behind is a very, very concentrated sugar solution. It doesn't have a lot of other benefits, people say, but it's an antibiotic. And it does have antibiotic, antibacterial, and antifungal benefits when it's applied externally, not when it's taken internally. So if you want to protect bacteria or protect the gut from bacteria, you can put honey on in a pinch and it will block the bacteria. It'll actually kill bacteria, but only when it's applied externally, not when it's eaten internally. When it's eaten internally or taken internally, the honey is broken down and all the calories of the sugars that are in it are released. And those of you who might be thinking that it's got other benefits like vitamins and minerals and other nutrients, not that much. It's not a significant source of vitamins and minerals. It is a very significant source of simple carbohydrates and calories, however. So you've got to be careful about using these substitutes. The same with agave syrup and some of these other sugar substitutes that people consider to be healthy. They're not really all that much healthier than sugar, because they are very, very concentrated sugar solutions, and they don't contain a whole lot of vitamins and minerals to supplement or to make them nutritionally superior to table sugar. So just bear that in mind. There's a lot of hype around these molecules as nutrients, or these alternative sweeteners as natural sources of sugar or natural sources of sweeteners. You've just got to be very, very careful about accepting that they are better alternatives. You need to pay close attention to what actually is going in. Again, take a look at the nutrition labels. Don't take my word for it. Take a look at the nutrition labels and see what is actually there on the nutrition labels, because the manufacturers are required under law to represent accurately what is present. That's what those labels are there for. So you might as well make use of them. The last type of polymer are the polysaccharides. These are the really long chains. Erase this. I'm not going to erase, I guess. See if we can try. Let's see if we can get the eraser. Can't get the eraser. Oh well. So these chains are essentially infinite in length. to all intents and purposes, they might as well be considered that way. There are three that are really important. So we have glycogen, which is how we store glucose in our muscles and in our liver. That's primarily where this is stored. And it's a form of instantaneous energy. So I'll put here, stored in muscles and liver. That's the source of instant energy. And flight or flight. Response. So when you get a release of adrenaline as a consequence of being wound up, and you get that instant rush of energy in the fight or flight response, that's basically the adrenaline response. It's the glycogen in the muscles particularly, but also in the liver, that's being broken down to glucose, which is being processed in the cell respiration pathway to provide you with that near instantaneous energy. And when the adrenaline goes away and you feel totally drained of energy, that's because you have literally burned through that glucose store in the muscles and in the liver. And until you restore that, you're going to feel significantly energy depleted as a consequence. Starch basically serves the same purpose as glycogen in plants. So it's a way of storing glycogen for short-term and long-term storage. And we'll talk about cellulose as a separate polymer, because it's the odd one out amongst these three, as you'll see. So when we take a look at a starch molecule, you can see that it comes in two forms: a linear unbranched long chain polymer of glucose called amylose or amylose, and then a branched form called a amylopectin. Both of them are just simple polymers of glucose. One is unbranched, one is branched. Think of the amylose as being a short-term storage molecule and a amylopectin as being a long-term storage molecule, because that's basically what they are. So there's two types of starch within the starch granules that you'll find in potato cells or in the chloroplast, which is an alternative place for storing starch. So the amylose is easily broken down because it's unbranched. The amylopectin, a little bit tougher to break down, but can be broken down nonetheless. Then you've got glycogen, which looks very much like a amylopectin, but it can be broken down very easily in response to the presence of adrenaline in the bloodstream. And so the muscles can free up the glucose in the polymer in response to the presence of adrenaline. That gives them access to the energy to do the fight or flight response to the presence of that adrenaline. Cellulose, on the other hand, is a different story altogether. And that's because when it comes to circularizing glucose, there are two different forms of circular glucose. So I'm going to jump past this slide for a minute to get to this one to show you what the two forms are. I'll go back to the other one in a minute. So when you circularize glucose, you get two forms, one called alpha glucose, which has the hydroxyl group on carbon number 1 pointing down below the ring. and beta glucose, which has the hydroxyl group pointing up above the plane of the ring. The alpha glucose is what is found in starch and forms what is referred to as an alpha 1,4 glycosidic linkage because it's linking alpha glucoses together. And this is the one that most organisms can break down. Starch is very readily broken down by most organisms on the planet because of the alpha 1,4 glycosidic linkage. In cellulose, however, it's the beta form of glucose that is linked together to form the polymer. And that gives what is referred to as the beta 1,4 glycosidic linkage. And it turns out that only two types of organisms can break this down. Some prokaryotes known as methanogens that belong to the prokaryotic group, the archaea, and they can break down cellulose, and fungi, which are the other major group of organisms that can break down cellulose. No other organisms routinely break down cellulose. It's because of this beta 1,4 glycosidic linkage that is undigestible by most organisms. But notice that it also changes the orientation of the hydroxyl group on carbon number 2 of the adjacent glucose molecules. And this changes everything. Because when you get the fibers of cellulose polymers adjacent to each other, those alternate hydroxyl groups can now form hydrogen bonds between the fibers. And it's these hydrogen bonds, trillions upon trillions of hydrogen bonds between the fibers, that give cellulose its unique property. Cellulose is what makes up the walls of plant cells. It's also what makes up wood. So not the cell membrane. This is a separate structure outside of the cell membrane, so it's outside of the cell proper, that surrounds all plant cells. So don't confuse the cell wall with the cell membrane. They are two separate structures, as you'll see in the next chapter. So not only does it make up cell walls, but it is the major component of wood, because wood is simply dead plant cells. So when a plant cell dies, what's left behind is the cell wall, and that's what makes up the wood and all wood products. So paper, anything that's made out of paper, anything that's made out of wood is made up of cellulose. And it also represents our dietary fiber. And so this is another reason why we have to be very careful about condemning carbohydrates, because dietary fiber, which is really important in our diet, a really good healthy diet contains a lot of dietary fiber. It's known to reduce the risk of heart disease and stroke because it inhibits the absorption of cholesterol. It inhibits the absorption of large amounts of fat. It provides bulk in our diet, and so we feel full without consuming a lot of calories because we can't process the fiber. We lack the ability to break down the beta 1,4 glycosidic linkage because we are not fungi and we are not bacteria that can break it down. So it turns out that this is undigestible to us. It makes up our dietary fiber. Really, really important. But again, it's not a simple carbohydrate. It's a complex carbohydrate that requires specialized digesting. And if you're incapable of digesting it, then you're not going to get any calories out of it. So this is why fruits and vegetables are an important part of our diet. because they contain healthy carbohydrates, carbohydrates that are important to maintaining our digestive health and our physical health. And so condemning carbohydrates out of hand is not a smart thing to do. These are really healthy carbohydrates. So when you have the choice between drinking a glass of orange juice and eating an orange, go for the orange, because there you're going to get a lot of complex carbohydrate in the form of the cellulose in the orange cells, whereas in the fruit juice, you're going to get a lot of concentrated fruit juice with a lot of concentrated sugars and virtually no fiber, because we like to take the pulp out of it, which is where all the fiber is. And so you're actually taking away what is nutritionally a very, very important part of an orange by taking the pulp out of it. And the same is true for any fruit juice, whether it be apple juice or grape juice, or whatever it is that you're referring to. Eating the whole fruit is much healthier than drinking the juice, because you are removing the dietary fiber as part of making the juice. And you get a very concentrated solution of sugars as a consequence. Yes, you may get some other nutritive benefits as well, but you're taking out one of the most important components as a consequence of that. So cellulose, wood, pound for pound, actually has the same tensile strength as wood, sorry, as steel. And so it all comes down to understanding the chemical nature of cellulose and what makes it so much different from starch, which is, again, a polymer of glucose, but is a soft white powder. Likewise, glycogen. Both of them are soft white powders. They're polymers of alpha glucose, however. Cellulose, polymers of beta glucose, formation of hydrogen bonds, changes the story completely. So if you're an animal that makes a living out of eating plant material, like a cow, you have to have in one of your stomachs, because a cow has two stomachs, a solution that contains prokaryotes and fungi that are able to break down the cellulose so that you can get the nutritional benefit. The alternative is to do what some large herbivores do, and that is to have a really large stomach, which means you've got to have a very large body to park that very large digestive tract, including the stomach, in. So an elephant gets really big, not because it particularly needs to be big, but because it's eating a lot of plant material. It needs to eat upwards of half its body weight every day in order to stay alive. It needs a big stomach to process that, so it needs to be a big body to put that in. And that is the reason why we think the really large herbivorous dinosaurs of the Jurassic got as big as they did. Some of them were over 100 feet from tip of nose to tip of tail. They were huge, some of the largest animals ever to live on the planet. But they were eating a plant diet that was very, very poor, because the plant material was very, very poor nutritively because of the conditions under which it was growing. And that meant they needed to eat a lot of plant material, and that meant they needed a big gut, which meant they needed a big body to put it in. And they seem to have been willing to experiment to see just how big you could get and still survive. And they did get really big as a consequence of this. So you have two ways of making a living out of eating plant material. Do what the cow does. Have a second stomach that has a population of organisms in it that can break down the cellulose, and chew your food twice. So the cow will take a mouthful of grass, chew it up, swallow it, goes into the first stomach. When that stomach is full, it regurgitates all that material, chews it a second time, and passes it on into the second chamber, where the microbes now take over and break the cellulose down so it can get a better yield from the cellulose, or get really big and eat lots of material, bearing in mind that you're not going to get a whole lot out of each mouthful. They're going to have to eat a lot to do it. So that's basically how herbivores get away with it. We as omnivores that were once herbivores have remnants of a large digestive tract. So if you still have your appendix, that appendix is actually a remnant of a second pouch in your digestive tract that was there to allow you to break down plant material when our diet was more plant-based than it is today. But as part of our human evolution, we evolved to, no sneeze, excuse me. We evolved a shorter digestive tract because it's easier to eat meat, and we also evolved fewer teeth in our jaw because we can cook our food now, which makes it easier to break down, so we don't have to chew tough material anymore, and we're also relying on getting nutrients out of food that's easier to digest than plant material. And so our appendix is a remnant or a vestige of part of our herbivore past, when our diet was more focused on plant material. That doesn't mean that we don't benefit from eating a lot of plant material. We do. We know that from research. But our digestive tract is now more in tune with eating a mixed diet of plant material and animal-derived material rather than focusing strictly on plant material. Even though you can live quite happily on a vegetarian diet or a vegan diet, that is personal choice. It's not a necessity as far as your body is concerned. Your digestive tract can process a variety of foods, both plant and animal derived, because it's that's how we have evolved to be able to process a mixed diet. But we still benefit from eating a lot of plant and plant material rather than eating a lot of animal material or animal-based food. There's one more modified carbohydrate that we need to talk about. This is called chitin. Some people say chitin, but I say chitin. I think that is the preferred pronunciation. It is a polysaccharide that is found in the exoskeleton of insects. It's made out of chitin. This is the chitin monomer. So you can see that it's a modified version of glucose where you've got this side chain containing nitrogen, a carbonyl group, and a methyl group. And this is linked together to form the chitin polymer that makes up the exoskeleton. Now, what's important about chitin is that we as animals can break down chitin. So insects are very much a possible part of a diet. And in some cultures, insects are part of the cuisine. I know from my own home country, from Australia, the Aboriginal folk eat a lot of insect material, grubs and insects, ants and so forth, as part of their diet. I've actually eaten a meal with some Aboriginal traditional hunters. They actually open up their culture to tourists so that you can participate in their food gathering activities and also sit down and eat a meal with them. And as part of the meal, we ate some insects, grubs of some moths, I believe they are, but the grubs are called witchetty grubs. They're about 4 or 5 inches long and about an inch in diameter, 3/4 of an inch in diameter. You can eat them raw or you can eat them cooked. They're a bit starchy when they're eaten raw. They have a nice almond flavor when they're roasted over the fire. I've also eaten honey ants, which is candy for the Aborigines, and I didn't get into the termites or the moths that they also sometimes consume. Depends on which part of the country you're in as to which part of the insects or which insects are on the menu. In Southeast Asia, worms and spiders and all sorts of insects are on the menu. And again, it's because we can digest the chitin. But they're also on the menu here in the United States. I remember when I was living in the Los Angeles area, going down into Los Angeles and finding lollipops, homemade lollipops being sold at stands that contained insects. in the lollipop as part of the unique nature of them. So yes, we can break down chitin. What's the whole point behind this, other than maybe making you think seriously about insects as part of the diet? It's that we can synthetically make chitin and we can spin it out as a thread, and we do so to use in medicine, specifically in surgery. Because we can digest chitin, you can make a surgical thread out of this and you can use it to sew up surgical wounds or other wounds where you have to close things deep below the skin and where you don't want to have to go back in again to take out the surgical thread. Because it's made out of chitin, over time it will be slowly digested, but not fast enough that it can't hold the tissues in place for the wound to start healing. By the time the chitin is fully digested, the wound, or that area of the wound, has already healed up. The surface stitches can be made out of other types of surgical thread because you can get access to those and you can remove those relatively easily. But in terms of going into the wound again, you don't want to do that because that just delays healing. So you use a chitin-based surgical thread that will be slowly digested away while the wound is healing. Kind of a cool trick when you think about it. But it takes advantage of the fact that, yes, we can digest chitin. So time to stop again because that's the end of the carbohydrates. So let's take a think about carbohydrates and take a look at these two questions. What is the general chemical formula for a carbohydrate? I didn't actually mention that in the presentation, but there is a general formula for a carbohydrate. And if you think about the name carbohydrates, you may actually be able to come up with it, but I'll show you what it is in a moment. What is the chemical formula for a monosaccharide that has 5 carbons? So if you stepped away for a moment, welcome back. If you are still here and puzzled by these questions, let's take a look. So what is the general formula for a carbohydrate? Carbohydrate, water, and that basically tells us what the general formula is. So the general formula is CH2O X. So any carbohydrate can be written as some multiple of CH2O. The number X obviously refers to the number of carbons and the number of hydrogens, and X times 2 gives you the number of carbons, number of oxygens, and X times 2 gives you the number of hydrogens. So that now leads us on into the second part here. What is the chemical formula for a monosaccharide that has 5 carbons? So if X is 5, which is what we're implying here, then it would be C5 and it would be O5, but we multiply 2 by X, which is 5, and so it would be H10. So a monosaccharide that has 5 carbons would be C5H10O5. That will be the answers to those two questions. So let's move on to the second class of macromolecules, which is our one class of non-polymer macromolecules, the lipids. These are referred to as hydrocarbons. So this is a carbon chain with lots of hydrogens. And as a consequence, these are hydrophobic, meaning they do not mix well with water. They are very water-fearing, hydrophobic. Remember, is the term that we used from chapter 3, our third presentation, to describe molecules that don't mix well with water. They are hydrophobic, and lipids, fats, are extremely hydrophobic because they are essentially pure hydrocarbon or near pure hydrocarbon. But they are not polymers. So we recognize two different types of lipids, lipids with fatty acids. And fatty acids are the hydrocarbon chains that are in question here. And then there are a family of lipids that lack fatty acids, and we'll talk about those at the end of this section. So let's take a look at what a fatty acid is and look at how that contributes to a lipid, or a lipid with a fatty acid, otherwise known as a fat. So a fatty acid, and I'm going to need to move this so I can see what's going on at the top for a moment. So a fatty acid, a lipid that contains fatty acids is sometimes referred to as an acyl glycerol or a glyceride. So these types of fats contain a glycerol backbone, which you can see here. So this is one component. So we have a glycerol backbone, and then we have this long chain of carbons, and this is the hydrocarbon. That is basically a lot of carbons linked together to form the chain with a lot of hydrogens attached to the backbone, and at this end we have a carboxyl group, the acid group, hence the name fatty acid, because it's got the acid group at the end. It's going to be the acid group and the OH group on the glycerol. Glycerol is an alcohol of glyceraldehyde, the glyceraldehyde sugar. So this is an alcohol of the glyceraldehyde, and that means the carboxyl group has been converted to an OH group. That's where we get the alcohols idea from. It's going to be the OH group and the carboxyl group that are going to form the link that links the fatty acid to the glycerol backbone. So what we wind up forming is what is referred to as a ester linkage. It is the linkage between the carboxyl group and the alcohol group as a consequence of taking the hydrogen off the alcohol group and the hydroxyl of the acid group. And this is our ester linkage. So that's the ester linkage that links the fatty acid to the glycerol backbone. Because the glycerol has 3 OH groups, you can link up to 3 fatty acids via ester linkages to the glycerol backbone. So this is what is referred to as a triacyl glycerol. These are body fats. So a triacyl glycerol or a triglyceride is a glycerol molecule with 3 fatty acids attached to it. Now, all of these fats, all of these fatty acids, can't see that, can you? Maximum number of hydrogens. I'll just leave it at that for the moment, I think. I was going to add something else in there, but I think I'll just leave it as that for now. So these fats are described as saturated fats because they have a maximum number of hydrogens attached to the carbon chain. And as a consequence of this, the fats are linear, or the fatty acids. And as a consequence of that, they pack tightly together and they tend to be solid at room temperature. So anything that has a large amount of saturated fat in it is going to pack tightly together and it's going to be solid at room temperature. And as soon as you start thinking about fats that are solid at room temperature, likely the one that comes to mind is butter. But there are other fats that are solid at room temperature too. Lard, which is the fat that's left over after you cook your bacon. If you cook bacon in a fry pan in the morning, when the fry pan cools down, you'll see that there's a white fat material in there. That's lard, that's bacon or pig fat. Another fat would be tallow, that comes from beef. There's one more that comes from beef as well, and I forget what it is. Oh, suet. Suet is another type of beef fat that's often used in cooking. Tallow is the fat that was once used for making candles before they started using beeswax and other non-animal fats, although beeswax is an animal fat as well. And it's solid at room temperature. So there is another one that's solid at room temperature. But notice the common thread here. All of the fats that are solid at room temperature, all the fats that are saturated, they're animal fats. And that's a fairly consistent theme that you should remember. Fats that come from animals are saturated fats, by and large. There are some exceptions, but by and large, an animal fat is typically a saturated fat. And it will form fats that are solid at room temperature. But then you've probably also heard that there are unsaturated fats, and unsaturated fats tend to be liquid at room temperature. So when you think about all the oils that you might use in cooking, like canola oil or plant or vegetable oil or corn oil or peanut oil or olive oil, the list of oils goes on and on and on. But one of the common threads in all of that is that they are plant-derived fats, because it turns out that plants make a percentage of their fats as unsaturated fats. So here you can see two fats. This fat is called, or fatty acid is called stearic acid. It's a component of butter, and you can see that it is a saturated fat. All the carbons are linked together by single covalent bonds represented by these lines. And then there are hydrogens attached to the skeleton everywhere else. So this is a saturated fat, and it is linear. This fat called oleic acid, or fatty acid called oleic acid, is exactly the same length in terms of the number of carbons as stearic acid. But it has one difference. It has a double bond between these two carbons, and you can see what effect that double bond has. It causes the carbon chain to bend. It's got a kink in it. And that kink means that this fat no longer fits nicely in and packs tightly against the saturated fats. It's going to occupy more space. And so these fats are not going to pack as tightly together as the linear fats of the saturated with saturated fatty acids. And so rather than being solid at room temperature, they're going to be liquid at room temperature. So the presence of a double bond changes everything. For one thing, it reduces the number of hydrogens by 2. So whereas if you looked at this and counted up the number of hydrogens, you'd reach a number. I don't know what the number is because I forget how many carbons there are in this chain, which would determine how many hydrogens are in. But what I can tell you is there are 2 less hydrogens on the carbon chain, which is why we say that it is no longer saturated, it is unsaturated. So unsaturated fats, by definition, contain a double bond and a reduced number of hydrogens, which means they've got the bend in here. And naturally occurring unsaturated fats are of the cis conformation. So you may recall that we talked about geometric isomers or cis-trans isomers in the previous presentation last week. This is where cis and trans now become important. Naturally occurring unsaturated fats are in the cis conformation, and that makes them liquid at room temperature. Okay. Now, this has posed a problem for food manufacturers. And the reason why is because the consistency of the food that we enjoy, like a soft, chewy chocolate chip cookie, or the classic French fry from McDonald's or anywhere else, all of those properties of our food are as a consequence of using saturated fats, animal-derived fats, because they give a solid fat at room temperature. That gives the soft and chewy chocolate chip cookie. If it was made from plant fats, it would be a lot tougher. It might be of the consistency of a hockey puck. That's the terminology that I've used in the past in describing the difference. And we all know that we would prefer the soft and chewy chocolate chip cookie. I mean, there's no mystery there. Or if you think about the classic McDonald's French fry, nice and crispy and golden brown on the outside and soft and fluffy on the inside. Well, the only way of getting that is to have really hot fat to deep fry them in. And the only way to get really hot fat is to have a fat that needs to be melted by heating it, and then will tolerate being heated to a much higher temperature in order to get that classic French fry. You can't do that with a plant fat because it's already liquid. If you try and heat it to the high temperatures that you can get an animal fat to, it's going to burn. We talk about the smoke point of a fat. That's the point at which the fat starts to burn. And oils have lower smoke points than animal fats, because an animal fat has to be heated before it turns into an oil, and then it can be heated even higher before it starts to burn, whereas fats that are already oils can only be raised to a limited amount of, in terms of temperature, before they start to burn. So what is a food manufacturer to do? Well, there is a solution, and the solution is to bubble hydrogen through the liquid fat. It's a process called hydrogenation. And when you hydrogenate an unsaturated fat, so the solution for food manufacturers was to bubble the hydrogen through the liquid fats and partially hydrogenate the fat or partially resaturate some of these double bonds. And what that would do was change the consistency of the oil. So that would be a little bit more viscous. It would start to approach the consistency of a solid fat, but it wouldn't be truly solid. That meant that it could still be heated a lot more. It would still give some of the properties of a solid fat in cooking, and it seemed to be the solution. The problem is with this as a solution is that not only does it partially hydrogenate some of the double bonds, but it also converts some of the double bonds from the cis form to the trans form. And this is where the problem really comes into play. So the reason why I've put there that no healthy level of trans fat has been determined is because trans fats in food are unnatural. In fact, trans fats in nature are essentially unknown. There are some exceptions to that rule. We know of a couple of organisms that do make small amounts of trans fats, but primarily unsaturated fats in nature are of the cis form, and the trans fats that occur in our food are the product of manipulation. They are synthetic, they're artificial. And because what happens, so I might as well draw a diagram of this. There's the cis. The conversion to trans is so you take the cis fats that have got the big kink in them and convert them to trans fats that re-straighten and behave more like saturated fats. And we don't know what a healthy consumption of trans fats is because trans fats are synthetic, they're artificial. They don't occur naturally in our food. And so what has been determined is that the recommendation is as little trans fat as possible, preferably zero. And so on January 1, 2007, legislation went into action from the USDA that determines all of these things that required food manufacturers to list the amount of trans fat in their food. But even before the legislation went into play, food manufacturers were already catching on and starting to reduce the level of trans fats and proudly advertising that their foods contained zero trans fat. The problem with this is that the food legislation provides the food manufacturers with wiggle room. There's a loophole. And you know that if a loophole exists, someone's going to take advantage of it. And the loophole is this, that you can label a food as having zero trans fat so long as it has less than 0.5 gram of trans fat per serving of that food. And it used to be that the manufacturers themselves decided what a serving of the food was. So if you were to buy, prior to around 2007 or thereabouts, if you were to buy a snack pack of potato chips in a vending machine, you might notice on the nutrition label that there might be let's use a round number, 100 calories per 1 ounce serving of potato chips. The problem is that that bag didn't contain 1 ounce of potato chips. It might have contained 1.5. It might have contained 3 ounces, depending on the size of the bag. But it was still a valid label because the food manufacturer decided that 1 ounce of potato chips was a serving. There was no rule that required them to use that. That's what they decided to use. And every food manufacturer had their own idea of what a serving consisted of. And so that's part of the problem. The other part of the problem is, notice, as long as it's less than 0.5 gram per serving, you can label it as being 0 trans fat. That means you could have potato chips or some other snack that contains 0.5 gram per serving of trans fat, or just under 0.5 gram, and it could legitimately say 0 trans fat. And remember, the recommendation is minimum amount of trans fat, 0 preferable. So how can you tell if there's trans fat in a food if they can legitimately label a food as having 0 trans fat with up to 0.5 gram per serving? You look on the ingredients label, because they cannot disguise the fact that there are modified fats present. You look for the term partially hydrogenated. If you see a partially hydrogenated fat as one of the ingredients, and they are required to list the ingredients, and they are required to list the presence of partially hydrogenated fats, then you know that there are going to be trans fats there, because the act of hydrogenating or partially hydrogenating the fat is going to produce trans fats. You cannot avoid that, and the trans fats will be there. Now, one of the interesting offshoots of all of this is Crisco. Crisco is a modified corn oil fat, and it is hydrogenated so that it is solid at room temperature, but it is a plant fat, but it has been manipulated. And the company that makes Crisco labels that there are trans fats in there, and they label how much trans fat is in there because they can't avoid it. But people like Crisco as an alternative to animal fats as a fat ingredient in cooking. But it is a hydrogenated fat, and it does contain trans fats. So you might want to think about that if you're thinking in terms of taking in trans fats versus saturated fats. Saturated fats are actually better for you than trans fats, as far as we can tell. But we need to do a lot more research and follow people's diets for a lot longer to find out the effect of trans fats on health long term. We know short term that there is an increased risk of heart disease and those sorts of conditions with trans fats. At least there's an indication, but it requires more long-term investigation to figure that all out. But the point here is, we know where trans fats come from. We know what creates the trans fats. We know that it's a product of man's manipulation of the existing cis unsaturated fats. And the only way to be really sure whether there's trans fat in a food or not these days, because of the legislation, is to read the ingredients and find out whether there are partially hydrogenated fats there or not. The other quick one that I want to talk about is the idea of using coconut oils and coconut fats as an alternative, as a healthy alternative to some of the other fats that are possible. Just bear in mind that if your coconut fat is solid at room temperature, that means it's got a lot of saturated fat in it. It's just like an animal saturated fat. And there are various types of plant fats that do have a high content of saturated fat, and those are typically fats that come from tropical plants. So palm kernel oil, palm oil, and a lot of coconut fat actually comes from tropically grown plants and contains a lot of saturated fat. You might as well eat the animal saturated fat. It is no more healthy than eating animal saturated fat. but there is a fad about using coconut fat as being somehow healthier for you. It's not. It's a fat in terms of calories. It contains just as much fat, or it's just as many calories as any other type of fat. Fat is fat no matter where it comes from. The problem with a lot of coconut fat is that it is solid at room temperature, which means that it is saturated fat, not unsaturated fat. Its plant derivation notwithstanding. So something more to think about. You've got to be really careful about capturing these fad foods and fad food usages, because sometimes that's all they are. They're a fad. There is no real nutritional reason why you should be using it. In fact, there in many cases are really sound nutritional reasons why you shouldn't be using it. And this is probably one where you need to be really cautious. There are saturated fats and unsaturated fats, however, that can be considered to be healthy. This is why we are encouraged to eat fatty fish from cold water environments, because there the fish are actually making unsaturated fats to use as an antifreeze. And these fats are known to be heart protective. They protect our cardiovascular system. So eating fish like salmon, tuna, other cold water fatty fish is encouraged. So eating these sorts of fish, preferably wild caught in a sustainable fashion, 2 or 3 times a week, is considered to be really healthy because of the fats that are present. But just remember, it's still fat, so you don't want to overdo it. Everything in moderation. We have a tendency to think, well, if I do something in moderation, that's good, but if I do more of it, that's got to be even better. No, that's not necessarily the case. And in many cases where things in moderation are beneficial, doing more is just not a good thing to do. And this would be an example of that. Fat is still fat. Now, there's one type of glyceride that we do need to pay attention to, and these are called phospholipids. They're diglycerides or diacyl glycerols. Let me show you a diagram because it really illustrates the point. So you have the glycerol backbone as usual. We have 2 fatty acids attached to 2 of the attachment points as usual. But in the 3rd attachment point, instead of a fatty acid, we have a phosphate group, hence the term phospholipid. And then attached to the phosphate group is another non-fat group that is also charged. So in this case, it's choline. So this is phosphocholine. I think that's the full name, phosphocholine. There's probably more to the name. But this is a phospholipid, and this is an animal phospholipid that you would expect to find in yourself. Why are these important? Well, if we look at this end of the molecule, particularly this region, but we can also add in the glycerol part, this is charged. And it's hydrophilic. The fatty acid region down here, this is uncharged. And it's hydrophobic. So you've got a molecule that contains both a hydrophilic region and a hydrophobic region. And molecules with this sort of conformation are said to be amphipathic. So I'm going to move this box down for a minute. You'll be able to see this in the recording. Amphipathic. This is a term that you need to become familiar with because we're going to use this repeatedly throughout the course of the semester. So amphipathic is the term that we use for a molecule that has both a hydrophobic and a hydrophilic region. So a molecule that is amphipathic will both interact with water and also interact with fats. So a detergent is an example of an amphipathic molecule because it's a modified fat. But in the case of the phospholipids, they're even more important because if you mix phospholipids with water, they will naturally form this phospholipid bilayer, where the fatty acid tails, the extremely hydrophobic part of the phospholipids, come together because they want to be together and exclude water. And the hydrophilic heads, the choline, the phosphate and the choline region in this case, interact with water. And they form this layer that separates water on one side from water on the other side. And this is the basis for a cell membrane. Not sure why it wants to keep doing that, but we'll live with it. Okay. So when we get to chapter 7, which we'll do in a couple of weeks, we're going to spend some more time talking about phospholipids and their role in cell membranes, because that's the chapter where we talk about the structure of cell membranes. So we'll need to remember the structure of phospholipids when we get there, but it's this spontaneous formation of the phospholipid bilayer whenever you mix phospholipids into water that forms the basis for all cell membranes, because they're the perfect way to separate water on one side, say the outside of the cell, from water on the other side, the inside of the cell, with a region that is extremely hydrophobic and therefore would prevent water from easily crossing from one side to the other, or anything that's soluble in water from crossing from one side to the other. And so that becomes part of our understanding of cell membranes, the fact that they're made up of these phospholipids, these amphipathic, meaning they have a hydrophilic region and a hydrophobic region that spontaneously form these phospholipid bilayers. So it forms the basis for every cell membrane, wherever we look. So more to come when we get to chapter 7 on that one. Then we have a group of lipids that do not contain fatty acids, and these are built on a steroid backbone, and cholesterol is the classic example of this. So this is cholesterol. And this is the steroid backbone. And we actually need to have cholesterol. We need it for two reasons. It is a component of all animal cell membranes, and we'll talk more about that when we do chapter 7. But it also is the precursor to the steroid hormones. So when you think in terms of estradiol and testosterone, cortisol, and other steroid hormones, these are all built from cholesterol. So much so that your liver every night while you're asleep is a cholesterol factory, churning out the cholesterol that your body needs, not only to make all the cell membranes of all the cells that it's making, and you wake up every morning with about 450 million new cells that you didn't have the night before. So obviously a lot of membranes being made, and making all the hormones that you need to maintain your body of the steroid form. They're all built out of cholesterol. So that's the reason why the liver is making cholesterol. It's a cholesterol factory. That's transported around the bloodstream in particles called low-density lipoproteins. And again, when we talk about membranes, we'll talk about the consequences of that, because there are some consequences to the transport of cholesterol. So that's another thing that we need to be aware of. And of course, we talked about estradiol and testosterone and the role of the functional groups when we talked about functional groups in the previous presentation last week for Chapter 4. So if you are unfamiliar with estradiol and testosterone and the role of functional groups, you might want to go back and take another look at that. But that's the end of the lipids, so time for another quick break and another concept check. So the question is, how does the structure of a phospholipid differ from that of a fatty acid or a triglyceride? So if you want to take a bathroom break or go and grab a drink, by all means, put me on pause. Otherwise, I'll stop for about 30 seconds so you can think through this question, and then we'll talk about the answer very quickly. So welcome back, no matter whether you took a break or you stayed here and thought it through, or maybe you even turned it off and walked away for a little while and came back to look at this as a second part. Sometimes feel like playing the Jeopardy music in here. I keep hearing the Jeopardy music playing in my head while I'm waiting for everyone, waiting to talk about the answer. Maybe I should think about that or something. Anyway, that's me thinking out loud. So the concept check question, how does the structure of a phospholipid differ from that of a fat, like a triglyceride? So what we need to remember is that they both start out with the glycerol backbone. So we need to remember that that's a similarity, not a difference. They both have fatty acids attached to the glycerol backbone. But in the case of a triglyceride, it's 3 fatty acids that are attached to the backbone, and that's it. So what we need to remember now is that a phospholipid has a phosphate group in the 4th location, and then attached to that this is some sort of charged group. So yes, we've got a slight difference in the arrangement, but that also means that we've got different properties. So this is strictly hydrophobic, and we know that this is amphipathic. Because you've got the uncharged fatty acid, the hydrophobic fatty acids, and you've got this charged area up here. So that's a quick review of the two, just so that you have an idea of the difference in structure. So onwards we go with the next group, and this next group is probably the largest group and perhaps the most important, depending on what perspective you want to take. These are the proteins and polypeptides. And in some cases, they can account, particularly in the case of plants, upwards of 50% of the material that's left behind after you remove the water, the dry components of the cells. So here's a list of some of the things that proteins can perform. This is a short list, in fact. There's a number of other things that we could put on here, but this is a short list of some of the more important things that proteins do for cells. And rather than go into a detailed discussion of this, in the interest of time, I'll leave it to you to read this section either in the textbook, or if you download the PowerPoint slides, you can read through these 2 slides. It just gives you some ideas of the various things that proteins can do based on that list on the first slide. So the couple of slides that outline that, you can read through those. What is consistent amongst all polypeptides and proteins is that they are polymers of amino acids. Now, as I mentioned at the very beginning, there is a temptation, and a lot of people do this, to use the term polypeptide and protein interchangeably. They are not the same. And at the moment, it's very difficult to explain why they're not the same, but that is coming. So I'm just going to use the term polypeptide for now, and we'll give you a more detailed explanation of the difference between the two when we start talking about the structure of proteins and polypeptides. So polypeptides are polymers, and they're polymers of the amino acids. And we've looked at the general structure of an amino acid before, but let's just remind ourselves what an amino acid looks like. So in the center is a carbon to which everything is attached. This is the alpha carbon, also known as the chiral carbon, or asymmetric. And so as a consequence of this, it's possible to get enantiomers of the amino acids with the, and all of the amino acids except one, glycine, exist as L and R enantiomers in the laboratory when we make them. When we make them synthetically, we make both the L and the R version, but life preferentially makes and uses only the L enantiomers. The R's do not exist in nature. Not naturally, at least. They can occur over time, but we don't need to go into that. Let me just leave it at that. L is the naturally occurring form because that's what life makes and that's what life uses. So the reason why they can exist as enantiomers, however, is because we've got this central carbon with 4 different potential things to be attached to it. So we have the amino group and the carboxyl group, hence the name amino acid. We have a hydrogen. And these are the common elements of all amino acids. What differentiates each amino acid is what's attached here as the R group, sometimes referred to as the variable or side group. And it is actually the R group that determines the chemical properties of each particular amino acid on the basis of what the R group consists of. So when we take a look at a diagram of the 20 amino acids, and by the way, you do not need to know the structures of these amino acids. If I ask any questions that relate to the structure of an amino acid, I'll provide you with a diagram of it. But I do not expect you to know the structures of amino acids. That being said, however, those of you who are going on to more advanced biology classes, because you're getting a biology degree or a science degree of some description. If you take some of the advanced biochemistry classes, you will have to learn the names and the structures of those amino acids, particularly if you're planning to go to professional graduate school, so medical school, dental school, vet school, so forth, pharmacy school. You will have to know the names of the amino acids and their structure. So that's something that you may be looking at in the future, but for this class, no, I don't expect you to know the names or the structures of the amino acids or their chemical properties. I will provide you with all of that information for any question where it's relevant. But what I want to point out here is that you can see the common framework of every amino acid. It's this blue purple box. I think you describe it as being purple. That's the common framework of every amino acid. But then notice that coming off the side is a different colored box, and that's the R group. And so if we look at the R groups for glycine and the other groups here, you'll notice that all of these are non-polar R groups. So all of these amino acids are non-polar and are hydrophobic. And these will tend to occur in regions of the polypeptide or protein that are hydrophobic. Why? Because these amino acids are there. So when we look at proteins and polypeptides, we recognize that there are different regions, or as we refer to them as domains, that have different chemical properties. So one domain may be hydrophobic, another may be hydrophilic. There may be an acidic domain, a basic domain, all sorts of different domains within proteins. What determines the nature of the domain is the type of amino acids that are there. What determines the chemical properties of each amino acids are the R groups. So these are the non-polar R groups, and any region or domain that contains these amino acids is itself going to be non-polar. These R groups are polar. So all of these amino acids are going to be polar. And so you would expect the regions that contain these amino acids or the domains to be polar themselves. Now, the last two groups of amino acids are polar, but they're also either acidic or basic, depending on the R group. So here are two amino acids that have acidic R groups, and you'll notice that there's a charge there, so that means they're also polar. So they're not just polar, they're acidic and polar. These are basic because they've got the amine group. And we said last week when we looked at the functional groups that the amine groups act as bases because they attract hydrogen ions. So here are R groups that have amine groups that attract hydrogen ions and have a positive charge. So they are basic. So these are charged, meaning they're polar, but they're also basic. So these are charged and basic. So we have non-polar amino acids, polar amino acids, polar and acidic, polar and basic. And the presence of these amino acids in different regions of the polypeptides and proteins determine the chemical nature of those regions, those domains. So when we start linking the amino acids together, we carry out a dehydration reaction, nothing new there, but we term the bond that forms between the adjacent amino acids as a peptide bond. These were named by German chemists who first deciphered the initial chemical characteristics of polypeptides and proteins. And so they named this bond that sits between adjacent amino acids as a peptide bond. And so how do you identify a peptide bond? You may ask, because you may very well have to. You look for this. You look for the residue or the remnant of the carboxyl group, which is going to be a carbonyl group once you take the hydroxyl group away. And that should be linked to a nitrogen with a hydrogen. That's the residue or remnant of an amine group. So here is a peptide bond in formation. Here's the hydroxyl group. Here's the hydrogen that's being removed. So notice in this case, it's reversed. We've seen previously there was a hydrogen here, a hydroxyl here. Well, now we've got it the other way around. So it really does, which you take from each unit does depend on the polymer and the monomers being used, but it's still the same thing. We take a hydroxyl and a hydrogen and form a new covalent bond, and that gives you the peptide bond. So there's a peptide bond there. Here's another peptide bond here. And you can see again, same conformation, the residue of the acid group in the form of a carbonyl, and the residue of the amine group in the form of this nitrogen and hydrogen. They are bonded together by the peptide bond. What that means is that when we start looking at a sequence of amino acids, a polypeptide, at one end, there's always going to be an unmodified amine group. and this gives you the amine end or N-terminus. At the other end, there's going to be an unmodified carboxyl group, which gives you the carboxyl end or C-terminus end. And new amino acids always going to be added with the carboxyl end. So consequently, a polypeptide is numbered from, let's clean that up a bit, I think. So we always number the amino acids in a polypeptide chain from the N-terminus to the C-terminus, because that's the order in which they're assembled. So when we start thinking in terms of polypeptides and proteins, we immediately have to start thinking in terms of structure. Proteins have very, very complicated structures, and it's that structure that gives rise to their function. So we've talked about structure and function being a pair of correlated features. If you know something about the structure, you know something about the function. If you know what function is to take place, that's going to tell you something about the structure. The two are very closely correlated or very tightly linked together. This is very, very true of proteins. Proteins have very, very complicated three-dimensional shapes. And so we have a number of different ways of representing those shapes. There's the ribbon model, which shows you a lot about the three-dimensional shape of the molecule and also shows you some of the internal features. But it doesn't show you anything about the amino acids that are present. You have the space-filling model, and these three diagrams represent exactly the same protein or polypeptide. So you have the space-filling model, which shows you each of the individual atoms and the space occupied by each of the atoms that compose the polypeptide. It's a little bit harder to see some of the intricate detail here, but it gives you an overall idea of the three-dimensional shape of the polypeptide. Then you have what is called the wireframe model, which shows you the covalent bond backbone, skeleton backbone of the protein, and then sometimes you can overlay in that the ribbon model so that you can see how the ribbon of the amino acids is constructed from the layout of the covalent bonds of between the atoms. So this is kind of a compromise between the ribbon model and the space filling model to show you more of the three-dimensional nature of the molecule. And pharmaceutical companies particularly pay a lot of attention to the wireframe model because it allows them to model the target enzymes or protein receptors that they might be trying to design a drug to target. And it allows them then to model both the naturally occurring molecule that would interact with the protein, and then also model their potential drug, the chemical that they're creating, that would potentially interact with the same protein, so that they can see if the two, if their drug is going to interact with the protein, does it actually interact with the relevant area of the protein that the naturally occurring molecule interacts with. So these are all models that can be drawn up using physical data extracted from the protein from a number of different techniques, and then assembled by a computer into one of these pseudo three-dimensional models. And there are computer programs that have been around for 25 years or more. I remember back in the, and I've got to think very carefully here, back in the early to mid 1990s, playing with computer models of this type as part of the research that I was involved in, modeling proteins that I had an interest in as part of my research. And it was relatively new to be able to do that on a desktop computer at the time. And we thought it was really cool that we could not only model the proteins on a desktop computer, but we could generate a pseudo three-dimensional model that would allow us to look at the protein in more fully in 3 dimensions, but we had to wear those funny 3D glasses that had the blue lens and the red lens that you sometimes still get at movie theaters when they're showing you 3D movies. We used to have to look at the computer screen with that to see the proteins in 3D, but we thought it was really cool at the time because it was, prior to that, you would have to book computer time on basically a supercomputer and go to a specialized computer facility to be able to model it. But by the time we get to the early to mid 1990s, you were able to do it quite easily with software on a desktop computer. Today, it's really easy to do. Your cell phone could do this. In fact, your cell phone can show some of these three-dimensional models. If you go searching around on the internet, you can find three-dimensional models of proteins that you can display on your cell phone. Anyway, moving on, let's talk about the structure of proteins. Where does it come from? Well, we recognize 4 levels of structural organization in polypeptides and proteins. The primary structure is simply the sequence of amino acids. Fairly simple to understand. The secondary structure and the tertiary structure and the quaternary structure or quaternary structure are a product of the primary. So as we move through the secondary, tertiary, and quaternary structure, we have to remember that this is all a reflection of the primary structure, the sequence of amino acids. So let's begin with the simplest, the primary structure. So here is a sequence of amino acids going from the amino end with amino acid number 1, all the way through to the carboxyl end with amino acid 127. And this is actually a protein that you possess. I believe this is the sequence for the protein that we're following through here called transthyretin. And this is the sequence of amino acids in one of the polypeptides that makes it up, it being made of multiple polypeptides. And you'll notice that each amino acid is given a 3-letter code. And if you know how to read the 3-letter code, you can read off the sequence of amino acids. I happen to know the 3-letter code. There's also a 1-letter code, which not too many people know. It was the original code. And it's more difficult to remember because some of the letters don't relate to the names of the amino acids. But the 3-letter code always relates the name to each amino acid, so it's relatively easy to remember. And I could show off and read off the sequence of amino acids, but I'm not going to. It's not important that you be able to do that. Just know that if you're going on to take more advanced biology classes and advanced biochemistry class, you'll learn the code. At some point. We all do. If you get into advanced biology classes, you'll learn the 3-letter code at some point. But that's the primary sequence. Now remember, that is what is going to determine the secondary, tertiary, and quaternary structure. And we'll talk about why that's important to remember when we get to the end of the protein section. So the secondary section is made up of 2 structures called an alpha helix and a beta pleated sheet. And both of these occur as a natural consequence of the sequence of amino acids. So when you get a certain sequence of amino acids, just because they link together in the way they do, they naturally fold into a helical structure. This is the alpha helix. So this is a natural consequence . So in the case of the beta pleated sheet and the alpha helix, it's the same idea. It's because you've got this particular sequence of amino acids in this particular region that it naturally folds into the alpha helix, or it will form the beta pleated sheet. What holds this together, because an alpha helix and a beta pleated sheet in a protein are important internal structures in the protein, so it's really important that they maintain. What holds these together is hydrogen bonds between an oxygen of a carbonyl group and a hydrogen attached to a nitrogen in an adjacent amino acid. So you can see here a hydrogen bond linking the adjacent turns of the helix together. And while a hydrogen bond is not particularly strong, remember, having lots of hydrogen bonds or even a few hydrogen bonds provides a stabilizing force, just as we saw in water, so we see it here. The presence of these hydrogen bonds stabilizes the alpha helix so that it doesn't fall apart. Likewise with the beta pleated sheet. If we looked at this pleated region, we would notice from the side that it goes like this. And so when we get a number of these side by side, we get our hydrogen bonds between the adjacent pleated regions in the protein. And these pleated regions could be consecutive in the sequence of amino acids. So these could be linked together in the sequence of amino acids, or they could be in completely different regions or completely different locations within the amino acid sequence. But because of the way the polypeptide folds around, they come side to side and cross-link with each other through hydrogen bonds. And you'll see an example of that in a diagram of the transthyretin protein coming up shortly. But this is the secondary structure. It's the natural formation of the helix, just the folding of the amino acids into the helix structure because of the sequence of amino acids, or the formation of these pleated regions that are linked together by hydrogen bonds just because of the sequence of amino acids. And they are stabilized by hydrogen bonds. And that's the other key piece here, stabilized by hydrogen bonds. So when you think secondary structure, you think alpha helix, you think beta pleated sheet, you think stabilized, or yes, stabilized by hydrogen bonds. The tertiary structure is where the protein takes on a far more three-dimensional shape. So the beta pleated sheets and the alpha helices are more planar, two-dimensional rather than three-dimensional, although that, I know that seems like a little bit of an oxymoron to say that. But the reality here is that it's at the tertiary level that the full three-dimensional structure of the polypeptide really becomes obvious. And in fact, in the transthyretin diagram, I can illustrate what I was meaning by the beta pleated, or the pleated regions sometimes being adjacent to each other and other times being in different locations. So here's a beta pleated sheet that we can look at. These two pleated regions are in fact sequential. You can see that they're linked there. But the third member is actually well separated. This goes off over here somewhere. This goes off over here. This pleated region is actually not sequential in the sequence of amino acids. It's located elsewhere in the sequence of amino acids. But because of the folding, the final three-dimensional folding of the polypeptide, these come next to each other and therefore are cross-linked by hydrogen bonds to form the pleated region. This is not unusual. This is actually quite common in the case of pleated regions. So what is it that holds this three-dimensional structure together? It's R group interactions. So The best way to think in terms of tertiary is that it is three-dimensional folding stabilized by R group interactions. So what sort of R group interactions can we get? Well, the simplest is just that they're hydrophobic. They want to be together because they're hydrophobic. So the polypeptide chain is forced to bend to accommodate these hydrophobic regions wanting to be with each other and to exclude water. And so you would expect to find these inside a polypeptide, away from the water around the surface of the folded polypeptide. The next interaction is a hydrogen bond, stronger than the hydrophobic interactions, but still relatively weak, but nonetheless, hydrogen bonds nonetheless. Now remember, these are hydrogen bonds between the R groups, so this is different from the hydrogen bonds that we saw in the secondary structure. If we want something that's a little bit stronger, well then the acid and base groups with their charges on them are now ions, and so they can form an ionic bond, the electrical attraction. So this would be even stronger than a hydrogen bond. And again, the polypeptide chain will bend to allow these to form, or the formation of these bonds will bend the polypeptide chain into its final shape. If we really want to lock that in, then we would talk about the disulfide bridge. And you remember that we talked about disulfide bridges in the last presentation with respect to getting a hair permanent or a perm, that the breaking and forming of disulfide bridges in the keratin protein that makes up hair allows you to change its shape. And here is where the disulfide bridge becomes relevant. It is that covalent bond that links adjacent regions of a polypeptide chain together. So when we carry out the chemistry of a permanent, we're simply manipulating these disulfide bridges that are holding the keratin protein in one shape and rearranging these disulfide bridges to hold it into a second shape, yet to be determined. So these are all the different types of bonds that you can get, or linkages that you can get between R groups that hold the three-dimensional shape of the polypeptide in its final form. Now, if at this point the folded polypeptide has function, has very real function in and of itself, we would not only call this a polypeptide, but we would refer to it as a protein. Because a protein is a structural unit or a functional unit in an organism. And so if the single folded polypeptide also takes on a specific function within a cell, it is not only a polypeptide, it is also a protein. But as we now know, many polypeptides, many proteins are made up of multiple polypeptides. And this is where the quaternary or quaternary structure comes into play. And there's a saying that I teach all of my classes that summarizes this, and it goes like this. All proteins are polypeptides, which is true, I know, but not all polypeptides are proteins. That is, So this is how we differentiate between polypeptides and proteins. If the polypeptide has its own function, then it is in its final folded form at the tertiary level, then it's also a protein. But if you have to combine multiple folded polypeptides together to form a functional unit, then the final functional unit is the protein, and the individual polypeptides are the components of that protein, even in their final form. But this is where the quaternary structure or quaternary structure comes in. So a really great example of a protein with quaternary structure is hemoglobin. And if we were on campus, I could show you a model that we have of hemoglobin that's been 3D printed. We got it from a commercial company, and it allows us to pull out one of the individual polypeptides and show you the components of the polypeptide that go together to make up the protein. But hemoglobin is made up of two polypeptides called an alpha globin and a beta globin. There are two alpha globins and two beta globins. Each of those globins folds around a ring structure called a heme group, and in the center of the heme group is an iron atom to which oxygen is attracted. And so it's the heme group that carries the oxygen, specifically the iron atom in the center. But these are then organized into the hemoglobin protein by assembling two alpha units and two beta units in this particular structure. So the alpha units and the beta units in their own right have no function. They can't transport oxygen. So they are only polypeptides. Alpha globins and beta globins are polypeptides. But once they assemble in this fashion, they become the polypeptide components of the hemoglobin protein, which has the function of carrying oxygen. So this combined unit of alpha and beta subunits represents the hemoglobin protein. This is what I mean by all proteins are polypeptides, but not all polypeptides are proteins. Sometimes you need multiple polypeptides to create the protein. And there are many examples of proteins that are made up of multiple polypeptides. So here's a summary diagram. Of course, the primary sequence is fairly simple to understand. It's just a sequence of amino acids. But then, of course, we get the alpha helix and the beta pleated sheet as a consequence of having a specific sequence of amino acids in a particular region of the polypeptide that naturally fold into either the helix or the pleated form, and it's stabilized by hydrogen bonds. The final 3D folding, remember, is the tertiary structure, and it's the product of R group interactions. So let's put in here R group interactions, just as a reminder. And then remember again, if it requires multiple polypeptides, and we have quaternary structure, multiple polypeptides forming the protein. So this is a nice summary diagram to bring all the bits and pieces together again about the structure of the protein. And it's visualizing the structure of the protein and understanding what goes on at each of the three levels that is probably the hardest part of understanding the protein. But if you can visualize this, if you can create models for yourself at home, then you can learn to visualize the structure, and that makes it a lot easier to understand what's going on at each level. So if we bear in mind that the secondary, tertiary, and quaternary structure of a protein are a function of the primary structure, the sequence of amino acids, it stands to reason that changing the amino acid sequence could have severe consequences as far as the structure of the protein is concerned. And we see this with a genetic condition that we're going to follow through this semester, or one of the two, and that is sickle cell anemia. So in normal hemoglobin, the beta and alpha units come together to form a hemoglobin molecule that exists as separate hemoglobin molecules within the red blood cell. In the most common change that causes sickle cell, the number 6 amino acid in the beta unit is changed from a glutamic acid, which is an acidic amino acid, to valine, which is a non-polar amino acid. That single change in amino acid changes the chemical property of this region, which changes the way the beta subunit folds. And as a consequence of that folding, that misfolding of the beta subunit, it allows the hemoglobin to come together to form large crystalline rods of hemoglobin. Rather than functioning as individual molecules, it forms these long protein chains, crystalline rod chains of hemoglobins. And as a consequence, you have a great reduction in oxygen carrying capacity, but it also triggers the reshaping of the red blood cell from its normal biconcave discoid shape to this characteristic sickled shape. And all of the problems that are associated with sickle cell disease are a consequence of that, and can all be traced back to this single amino acid change that unfortunately happens to be in a key region that affects the overall folding of the beta globin subunit. And that has all the knock-on effects can be traced back to this single amino acid change. So sometimes changing a key amino acid in a key region affects everything. But this just reinforces the fact that the primary structure, the sequence of amino acids, is the origin of everything else. The secondary structure, the tertiary structure, and ultimately the quaternary structure. And this is a great example of that. But it turns out that proteins are also very sensitive to environmental conditions. And when you alter the environmental conditions significantly enough, you can change the structure of the protein. And of course, as soon as you change the structure of the protein, it loses its function. So when you heat a protein, you cause it to denature, which is the technical term for what's happening here. It's losing its structure, it's denaturing. It turns out, however, that altering the pH can do much the same thing. So when you cook an egg, the white turns white. Excuse me. So when you crack an egg open, you've got a raw egg. The egg white is actually transparent. It's a very concentrated solution of a protein called albumin. When you heat it, the albumin loses its shape, and it changes the egg white to something that was transparent, from something that was transparent to something that is now opaque. It's white in color. And you can't uncook it. This is an irreversible change to the protein because it's been denatured. What is interesting is that you can do the same thing by altering the pH. So if you add lemon juice to a raw egg, you get the same thing. The white turns white. So if you want to check this for yourself, sacrifice an egg, break it into a glass, take a really juicy lemon and squeeze the lemon juice into the glass. And you'll see that the white of the egg will turn white, just as if you'd cooked it. And in fact, you see that happening in some dishes. So if you are a fan of ceviche, you'll know that ceviche is made by not cooking the seafood, but by adding lemon juice and lime juice. And there again, you're affecting the pH. So you are technically cooking the seafood, you're denaturing the protein of the seafood by changing the pH. And pH can also have the same effect as cooking. So sometimes we cook to kill off bacteria. Altering the pH can also have the same effect. But I wouldn't count on that with the seafood. Make sure that you use quality seafood when you make ceviche, because you're not going to cook it, you're just going to alter the pH. And that, depending on what the condition of the seafood is, that may or may not be sufficient. The ceviche is traditionally not cooked. It's the pH that is changed by adding lemon juice and lime juice that makes the change. We can alter the salt concentration of the solution. And that changes the shape and therefore the solubility of the protein. And this is a common way of collecting proteins from solutions. You might extract the proteins out of an organism using a water-based solution. Now you've just got a collection of proteins. If there's a particular one that you want, you can isolate that by adding salt, and that changes the shape of the proteins and they come out of solution. And different proteins are affected by different concentrations of salt. So you can play with the concentration of salt to get rid of the proteins you don't want and to collect the protein that you do want. And this is a very classic manipulation that we do in biology research labs. And then chemicals can also change the shape of molecule of proteins. So things like ethanol, that's the alcohol in wine and spirits. I'm trying to think of some chemicals that are not acidic or basic, because that's obviously changing the pH. Alcohol's a classic one. If you want to see the effect of alcohol and denaturing proteins, you can't actually see the proteins denaturing, but you can see the consequences of this. Put a little bit of your favorite clear adult beverage. Vodka's a good example, a good one for this. In the bottom of a glass, go out into the garden, find an earthworm, clean it off and drop it into the alcohol, and it will die instantaneously, because the alcohol denatures the proteins that are working as enzymes. That shuts down the earthworm's biochemistry, and it's dead. Alcohol is actually a cellular poison, and it's just as poisonous to you and I as it is to the earthworm. The only difference is, because we have a larger body mass, we can tolerate more alcohol than an earthworm. But if we raise our alcohol concentration to the same concentration of alcohol that killed the earthworm, it will kill you and I just as much as it does the earthworm. It's called alcohol poisoning. It's something that unfortunately young people are very prone to, particularly at college and university, where fraternities and sororities often have alcohol drinking contests. And if you drink too much alcohol in a short enough period of time, your alcohol level goes so high that it poisons your cells and it will kill you. Your body can only eliminate alcohol at a slow rate. And so if you overconsume the alcohol and you raise the alcohol concentration to toxic levels, you will die. It's as simple as that. So you need to be very, very careful. And it's surprising how little alcohol can affect people. So the recommendations for alcohol consumption, and this is what surprises most college-age students, for an average woman is 1 alcoholic beverage, standard alcoholic beverage per day, and that is a glass of wine. Not a full glass of wine, but about 5 or 6 ounces. a shot glass of spirits, or a 12-ounce glass of beer. They all have about the same equivalent alcohol concentration. For a man, it's 2 standard alcoholic beverages. And the reasons why is because men typically have a slightly larger body size, or a larger body size than a woman. Of course, tolerance levels can be increased as well, but that doesn't, in terms of the amount of alcohol you can consume without getting drunk or feeling the side effects, but that doesn't change the toxicity. That never changes. It's the percentage of alcohol that becomes the toxic feature, not the amount of alcohol that you can tolerate and not get drunk or feel the effects of it. It's the percentage of alcohol that will kill you. We do know that misfolding of proteins is associated with a number of diseases. There's a particular type of misfolded protein called a prion that cannot be destroyed by heating, by cooking. And it's the presence of those misfolded proteins that cause other related proteins to misfold themselves and create a diseases. So mad cow disease is known to cause this. The human equivalent called Creutzfeldt-Jakob disease is also produced by prions. Scabies in sheep is called by this, and a human condition that was prevalent amongst the cannibals of New Guinea, and to some degree still exists in some locations where tribal culture says that it is an honor, or it is honorable when a relative dies to consume their brain tissue, called kuru, K-U-R-U, kuru is its name, and the transcription service is having trouble dealing with it. It was very prevalent in New Guinea. Occasionally there's an outbreak of it, but the consumption of human tissue has been banned. Cannibalism is illegal in New Guinea and has more or less been wiped out. But in some isolated regions, the eating of dead relatives' brains is still practiced as a way of honoring the dead relative, and it is what transmits the prion that causes kuru from dead relative to living relative, and just increases the number of dead relatives, I guess. But authorities are working really hard to try and convince all the cultures that there is a way of stopping kuru, and it is simply don't eat the brain tissue of your dead relative, particularly if they died of kuru. And there are ways of making sure that key proteins fold correctly. This is not actually in your textbook. This was included in previous editions of the textbook, then have taken it out. I've left it in my PowerPoint, but it's not essential that you know this. But there are these protein complexes called chaperonins that actually serve to make sure that proteins have an environment in which they can fold correctly instead of misfolding at times. And this is used particularly to protect key proteins that are important to the normal function of cells, particularly in conditions that are more hostile to the ongoing existence of cells. So high salt, high temperature, low water, these sorts of conditions, the chaperonins become really important in maintaining the normal folding of proteins. So we're getting to the end of this chapter, I promise. This is a long chapter, but it's because this is a fairly energy-dense chapter, and there is a lot of interesting side information that I can add in here that I think makes the basic principles a little bit more relevant and ties it a little bit more to your interest as a potential health science student, health professional in the future. We've only got one more set of polymers to look at, and that's the nucleic acids. So let's take a quick break. Here's another concept check. what type of chemical bonds hold together the secondary and tertiary structure, respectively, for polypeptides? Okay, so secondary, oops, right on the right screen. So secondary, it's all about hydrogen bonds. And it's hydrogen bonds between the amino acids. For tertiary, it's hydrophobic. Hydrogen, ionic, and covalent bonds. And in this case, between R groups. That's what holds the tertiary structure together. So secondary, it's hydrogen bonds between the actual amino acids themselves, not the R groups. In tertiary, it's hydrophobic interactions, hydrogen bonds, ionic bonds, and covalent bonds between the R groups of amino acids. So let's move on to the last section. Hopefully this won't take too long. We'll see how we get on. So the nucleic acids are the last form of polymers. These are made up of nucleotides as the monomers. We recognize two large polymer types in cells, deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. So DNA is the genetic material. It's the information containing molecule that's passed on from one generation of cells to the next, or from one generation of organisms to the next. RNA is involved in extracting the information out of the DNA in a form that can be used to assemble polypeptides and proteins, which is typically what the information in the DNA codes for. It contains coded information for the assembly of polypeptides and proteins. And so RNA acts as an intermediate, specifically a type of RNA known as messenger RNA or mRNA, but there are other RNAs that are involved in the process as well. So tRNAs, rRNAs, so tRNA, rRNA, siRNA, miRNA, so they all have roles in gene expression and regulation. And in fact, we're starting to get the impression that RNA molecules may be central to our understanding of gene expression and regulation because of their central role. And we're finding a whole host of tiny little RNAs like the siRNAs and the miRNAs that play very active roles in regulating gene expression. We don't get into that in this class. But again, if you're going on to take more advanced biology classes, and particularly biochemistry classes, you're going to learn a whole lot more about those little RNA molecules and their role in regulating gene expression as you take those classes. So this is the flow of information that we recognize, sometimes referred to as the dogma of molecular biology. So the information is stored in the sequence of nucleotides in the DNA. That is copied as an mRNA molecule, a messenger RNA molecule, and it's only part of the DNA, the functioning part of the DNA that is copied. That then passes out to the cytoplasm, where it is read by the ribosomes, and they use the information in the messenger RNA to assemble polypeptides. And so there are small RNAs involved in processing the RNA that's copied from the DNA to form the messenger RNA. There are RNA molecules in the ribosomes, those are the rRNAs. There are tRNAs that are involved in assembling the amino acids together, that actually carry the amino acids to be assembled. They're all involved in the synthesis of polypeptides and proteins. And then you've got the siRNAs and the miRNAs and SNPs and SNRPs and SNOs. These are all different types of RNA molecules that are involved in regulating genes. So that the nucleic acids do a number of different things. We're going to see a couple of metabolic roles for nucleotides as well, right at the very end. So when we look at the structure of a nucleotide, we find that every nucleotide has a sugar, two different sugars depending on the type of nucleic acid that we're looking at, a phosphate group that's attached to what is referred to as the 5' carbon of the sugar, and then an identifying molecule called a nitrogenous base that is attached to the 1' carbon of the sugar. And it's the nitrogenous base that determines what nucleotide you're looking at. These are assembled into a polynucleotide or nucleic acid strand. And the polynucleotide consists of a bridge between the 5' carbon of one nucleotide and the 3' carbon of the adjacent nucleotide, and that bridge consists of the phosphate group. So the phosphate group attached to the 5' carbon of one nucleotide bridges to the 3' carbon of the adjacent nucleotide. So why the prime designation for the carbons in the sugar? Simply because the nitrogenous bases themselves also have carbons. And so the decision was made by the organic chemists that first started to decipher all of this, to give the prime designation to the carbons of the sugars to separate them from the carbons of the nitrogenous bases. So the carbons on the sugars are called 1', 2', 3', 4', and 5'. For our understanding of DNA and RNA, the only two that we really need to remember is 5' and 3'. And the reason why is because on every polynucleotide, there is an unmodified phosphate group that's attached to the 5' carbon of the nucleotide at that end, and this becomes the 5' end. At the other end, there's an unmodified hydroxyl group attached to the 3' carbon of the nucleotide at that end, and that becomes the 3' end. And so we can recognize a 3' and 5' end of every polynucleotide. And that becomes important in understanding the function of both DNA and RNA, recognizing that there are ends and what that indicates about the molecule. Now, as I said, each nucleotide has a sugar based on the type of DNA, the type of nucleic acid that it's part of. So for DNA, it's deoxyribose, and for RNA, it's ribose. And the difference is the H, or the hydrogen, on the 2' carbon of deoxyribose, whereas it's a hydroxyl group on the 2' carbon of ribose. So here it's missing an oxygen, so deoxyribose. So this is in the nucleotides of DNA, this is in the nucleotides of RNA. So that's one of three differences between DNA and RNA. Different sugar. So there are so number one, different sugars. So that's something to remember. Number two, Everyone's heard of the letters A, G, T, and C. Those are the ones that are found in DNA. So adenine, guanine, cytosine, thymine. Those are the DNA nitrogenous bases. And they symbolize the, actually, you know what I ought to do, seeing as how there's already been a color coding given here. So in DNA, it's adenine, guanine, cytosine, and thymine. So those are the nitrogenous bases on the nucleotides of DNA. In RNA, the T nucleotide is replaced by the U nucleotide called uracil. And when you look at thymine and uracil, you'll notice that in thymine, there is a methyl group here that is missing in uracil. And that doesn't affect the interaction of thymine and uracil with their complementary base, which happens to be A. They can still complement with A because the methyl group is not involved. So it is a legitimate replacement. So you also notice that the nitrogenous bases are divided into single ring bases called pyrimidines and double ring bases called purines. And a colleague of mine that I was watching teach actually used a statement that I'd never heard before, and I mentioned this to my students just so that you can remember the two. You cut purines. So if you're struggling to try to remember which of the, sorry, you cut the pyrimidines. So even I make the mistake at times. So let's take out purines. We don't cut the purines, we cut the pyrimidines. Even though I've got it sitting right in front of me, we cut the pyrimidines. So cut cytosine, uracil, thymine. And those are the ones with a single ring structure. Purines have a double ring structure. Okay, so I don't consider this to be absolutely essential information to know in terms of being able to recognize pyrimidines from purines. But if you're trying to remember it, there's a statement. Though, again, those of you who are going on to later upper division biology classes, particularly biochemistry classes, you will need to know the difference. And you may even need to know the difference in an A&P class. I don't know for sure because I don't teach A&P, but it's possible that you'll need to know it for A&P at some point along the way. So there's two of the differences, different sugars, different nitrogenous bases. The third sugar, or the third difference, sorry, DNA is double-stranded. That's horrible writing. Let's see if I can do this better. So there are the three differences. Different sugars, deoxyribose in DNA, ribose in RNA, different nitrogenous bases. So thymine in DNA, uracil in RNA, and then DNA is always double-stranded, RNA is single-stranded. So there's the three differences. The sequence of nucleotides is how we store the information in DNA, and the sequence of nucleotides are read in groups of three called a codon. So the groups of 3 nucleotides are still present in the DNA molecule, but we don't refer to them as codons in the DNA. We refer to them as codons once we've created the copy of the gene in the messenger RNA. But there are still groups of 3 nucleotides in the DNA representing each of the amino acids. So the challenge, once it was recognized that DNA was the molecule of inheritance, was to try and figure out what the coding was. And once it was realized that it was grouped of 3 nucleotides, to try and figure out what all 64 possible codons actually meant. And we do now have the genetic code fully deciphered. It took about 12 years or so, from 1953 through to around 1965, 1967, somewhere around there, to fully decipher the genetic code. But when we get into chapter 16 and particularly chapter 17, we'll talk more about the genetic code and how that information is arranged, how the codons are organized. So when we think in terms of DNA, as I said, it is double-stranded. So here is a diagram of the DNA molecule, and you can see the double-stranded nature. But what is also relevant here is that the 2 strands complement each other, is the way we say this. So A complements T, or pairs to T. So you can see that where in one strand you've got an A, you've got a T in the other strand. And G complements C. So wherever you've got a C in one strand, you've got a G in the other strand. But they may complement each other, but they are anti-parallel. So what does that mean? That means that if you look at one strand and it's running 3' to 5' in one direction, the complementary strand runs 3' to 5' in the opposite direction. So if we follow this strand through, it's got its 3' end there, and we follow this one through, you find the 5' end down here. But if we look at this one over here, the complementary strand, its 3' end is down here, and its 5' end is actually hidden behind the box up here. Let me pull that down for a minute. So the 5' end is up here on this other one. So 3' is down the bottom, its 5' is up here. So they're anti-parallel to each other. The complementary nature of the 2 strands then provides us with a simple explanation for how it's copied. And when Watson and Crick published their paper, they actually hinted that it was copied in a what is referred to as a semi-conservative fashion. And we now know that that is correct. It only took 3 years to figure out that Watson and Crick were correct. So Watson and Crick published in 1953, and the paper that confirmed semi-conservative copying was published in 1956. And again, we'll look at that in chapter 16 in more detail. So I'll put in here that DNA replication is semi-conservative. Each existing strand acts as a template to create the missing strand. Using the base pairing rules. So when we say that the two strands complement each other and that A complements T and G complements C, the other way of expressing this is to say that A pairs with T and G pairs with C. So these are also referred to as the base pairing rules. That's a little bit more information about the DNA molecule. Yes, we go into a lot more detail about the molecule when we get to chapter 16 and 17, but as I said at the beginning of this section, it helps to give you a little bit of background information now because that gives you a solid foundation for when we get to chapters 16 and 17. RNA is single-stranded. In some cases, it is possible to get complementation in a single RNA molecule. So when we look at translation, the process of assembling polypeptides using information messenger RNA, we'll encounter a molecule called tRNA. And tRNA actually folds into a complicated three-dimensional shape because particular regions of the RNA molecule actually complement each other, and it folds over and folds into this three-dimensional shape because of that complementation. So again, we get the replacement of T with U, so you get an AU pair rather than an AT pair. So here you can see the folding of a tRNA, and you can see the complementation between certain regions that allow it to form a short span of double-stranded RNA, but in reality, this is a single-stranded RNA molecule that happens to fold because of the complementation, whereas DNA is always double-stranded because the two strands are fully complementary to each other. Then there are special roles for nucleotides in terms of energy. So we'll talk more about ATP and NADP plus and NAD plus when we talk about, in the case of ATP and ADP, when we talk about energy transformations in chapter 7 and cell respiration and photosynthesis in chapters 9 and 10. Sorry, energy manipulation in chapter 8 and cell respiration and photosynthesis in 9 and 10. And we'll talk about NAD plus and NADP plus when we talk about cell respiration in chapter 9 and photosynthesis in chapter 10 respectively. So more to come about these, but I just want to make you aware of the fact that this is another highly specialized role for nucleotides, and these are related to the equivalent RNA nucleotides. So this is adenine triphosphate and adenine diphosphate and adenine monophosphate. So it's the A nucleotide of RNA, and this is nicotinamide adenine dinucleotide. So again, it's related to the RNA nucleotide, not the DNA nucleotide. So one more concept check, and then we're done. So if the nucleotide sequence in one polynucleotide strand of a DNA molecule reads 3', C, T, T, A, A, G, 5', what will be the nucleotide sequence in the complementary strand? If you wanted to specifically label RNA with a fluorescent tag, what compound could you use that is or are unique to RNA? So let's do this one immediately. So if we've got a 3' to 5' sequence of C, T, T, A, G, 5', then remember the strands are not parallel, they're anti-parallel. So it's going to go 5' to 3' in the opposite direction. C will pair with G. T pairs with A, T pairs with A pairs with T, A pairs with T, G pairs with C. So in fact, these are mirror images of each other. And this is in fact a sequence of nucleotides that's recognized by a special enzyme called a restriction enzyme. And it's a particular restriction enzyme called ECHO-R1. And it will cut DNA, and it will cut this particular DNA right through here. It recognizes that sequence and cuts it right there. This is a way the bacteria can protect themselves from foreign DNA, which they like to take up from their environment, ironically enough. So if you wanted to specifically label RNA, what could you use as a fluorescent tag? Well, remember, RNA doesn't have T, it has U. So we could use U. Or if we wanted to use a sugar, we could use ribose, because these are two of the molecular differences between RNA and DNA. The U nucleotide is unique to, or the U nitrogenous base is unique to RNA, and the ribose sugar is unique to RNA. So either of those would work if we wanted to uniquely label RNA. So that brings us to the end of this section. It has been a long recording, I'm sure. I don't know how long it is, but it has been quite long. But I will post this as soon as the transcription is done so that you can look at it. The next presentation is going to be on chapter 6. That will be for later this week. I should have that posted sometime on Wednesday for you. In the meantime, if you have any questions or concerns or comments about anything that has been posted with this presentation or for the first 4, because remember, the first 4 are what turn up on the exam late at the end of this week. So you might want to come and ask questions in office hours. But by all means, if you have any concerns, questions, or comments about anything that's been in this presentation, by all means, come and see me. Otherwise, I will see you again for the next presentation, and that will be about chapter 6. Bye for now.